I will look into the item of evidence-based treatment and CCT indicated and performed outside the pharmaceutical industry. How to change this? Okay. The way of performing evidence-based treatment is actually based on what they call controlled clinical research methodology and controlled clinical trial. And everybody, even I, when I started in my career as a medical doctor and researcher, I think that controlled clinical trial was a very old science until I actually obtained that it was not actually so old, it was quite new. Even though we have a sort of history, and I think it's very important for all of you to know the history, the background for this basic item of evidence-based medicine, which we are talking about everybody, and that's controlled clinical trial today. Actually, it started, the first time I can get what we can see a clinical trial published. That was back in 1537, and that was Amosius Peer who reported that from treatment of wounds after battles. In Europe, they were starting to fight each other with sword, and the medical doctor, mainly the surgeon, have to take care of the wounded patients. And the usual way of treating these wounded patients was by heating oil. They put heating oil on the wound, and you know what's happened then. There was one big battle in France, and there was a lot of victims, and a lot of warriors which was hurted, and the doctors had a lot to do, and they ran out of heated oil. So they had to find another way of treatment, and they had to find it there and then. And the only thing they could do was to run around to the farmers and get some eggs and some mixture and make that together and put that on the wound. The day after, they have to inspect the patient and they see what the results were. And they found that the patient which had got this new treatment, this egg treatment, actually performed much better than the one who got the old one, the heated oil. And all of you know the reason why that. Since then, this was a clinical trial. And it was the next clinical trial, which I actually have seen it's observed, was from the East Asian Shipping Company. The big problem with the sailors was scurry. And it was common that around 55% of all the sailors died on the trip from Asia to Europe. And there was a big problem. And there was a young doctor. He had some thoughts about that. And he made a clinical trial, so to say. And that was the first, what I can call a planned clinical trial. And he had this company, they have 4 vessels going from India, and he loaded 2 of these vessels with citrus orange, and so on. And all the sailors had to eat at least 1 orange every day during the trip to Europe. And the other 2 vessels was going without. And when they come to London, they could count the results, the hard results, the number of dead. And they found that the number of dead in the 2 vessels with a usual way of giving foods was around 55%. But in the 2 with the orange, nobody was dead of scurry. You know, it's not necessary to be a statistician to see the difference, huh? But still, you know what's happened with this researcher? They fired him. So welcome to the club. And the worst thing is that he was actually the first to detect a reason for get a treatment for this scurry, or prevalence against this scurry. He couldn't explain the mechanism action because they don't know about C vitamin at that time. But it should take 150 years. 150 years, my friend, before somebody actually obtained this, and that was James Lynn. He found this note from this fired doctor, and he had to treat them. And he reported that in 1747, a paper in the London School of Medicine. He published that he had treated 12 patients with scurry with different kind of oranges, and reported a fantastic effect on them. Nothing happened. Nothing happened. Norway is a big nation with regard to sailing, and they had this same problem. And first, in the last part of the 1980s, they started actually to look into the possibility of giving oranges and such kind of C vitamin to the sailors. It took 250 years from this was reported first time to it actually take care of the disease. Think about that as a clinical pharmacist. If you found something new, don't think that this will be taken into account at once. It is a lot to fight it through that part and get it registered as such. The second controlled clinical trial, which actually is a sort of placebo-controlled trial, was reported by James Highgarten, and he was reported that in treatment of rheumatoid arthritis. And he called it tractoring, means that he was tracking out the joint. Huh? Fantastic. Huh? And he reported that there was a huge placebo effect of that, I can imagine, because if I were a patient, I want to have placebo. But anyway, it was a sort of placebo-controlled trial. The first time randomization was actually introduced was as late as in 1921, and that was what an unknown person called Ray Adam Fisher. You know who that is? Ray Adam Fisher? It's 3 of them. God heaven, it's good, it's not more. 3. Have you heard about t-test? Have you heard about analysis of variance, ANOVA? Have you heard about covariance? That's the 3 of them. This is the Fishers. And it was the grandson. In 1921, he introduced randomization, not in medicine, but in agriculture. And that was the first time that was actually registered. And the second time it was registered, was randomized clinical trial, was Ambrosius when he looked at pulmonary tuberculosis in 24 patients. All these studies was very, except for this battle study, it was actually a lot small studies. It's not a big scale studies, but that was the background. Remember, this is a background for all the treatment which is performed in a randomized clinical trial way. And the one which is quite famous was by Deal against the common cold. And of course, this had to be summarized in a way, and they summarized that. And the person who summarized that was Sir Austin Bradford Hill. He published in his book, which is called Statistical Method in Clinical and Preventional Medicine in 1962. And he is said to be the father of controlled clinical trials. He is the father of what we today call evidence-based medicine. So from that, we are starting to develop. And of course, the part which saw the possibility in this new science was pharmaceutical industry. And because this was a way of proving the effect of the new drug, looking into adverse events of the new drugs and so on, and they could actually prove that the new medicine which they put on the market was have a certain effect, and also additionally could report adverse events. And they was alone for developing all kind of methodology, all kind of routines, all kind of way of performing clinical trials was actually put forward by the pharmaceutical industry. But in 1980s, something happened, because then there was some researcher who found that this is actually not only related to this trial of pharmaceutical industry, it had to be also performed in another way, in a way which we can prove that the treatment which we are giving in subpopulations or at hospitals actually needed such kind of treatment. And then it was divided in 2 parts. We have this pharmaceutical industry performing large-scale studies with thousands and thousands of patients, costing millions, large-scale studies, and the only mainly the design which they are using is the parallel group design, and they need a lot of patients. But on the other side, we have the university hospitals, or the hospitals, the research institution, which also want to perform clinical trials. But they didn't have the economical power to stand behind the pharmaceutical industry. We don't have so much money. And additionally, we don't have all this patient which is needed for running clinical trial on the same field as the pharmaceutical industry. So what we need is to perform small-scale studies, and in order to ensure that these small-scale studies have the evil power that the huge pharmaceutical industry, we need something, and what we need is actually more powerful design or more smartness in the way we are performing the studies. If you look into the treatment, when you shall treat the patient, well, then you have a basket. And this basket has several rooms. We have one room with surgery. We have one room with pharmaceuticals. But we have one room with manual therapy. We have one room with physiotherapy. We have one room with Chinese traditional medicine. We have a room with homeopathy. And we have a room for others. All this here needs or is claiming the same. We need evidence-based background for performing the treatment. We cannot, if I have a patient, I cannot treat the patient whatever I want. I have the responsibility, and I need to know that the way I'm doing that is giving the treatment in the best way for the patient. So we need evidence-based. And of course, based on the pharmaceutical industry, the best evidence-based treatment so far ever, it is in pharmacy. There's a lot of every time you come a new drug on the market, it is a huge documentation with regards to the efficacy, indication, adverse events, and so on. And that's nice. But when you're giving this new drug to a certain hospital, the background which is made there is made by a huge amount of patients all over the world. And to which extent will your small group of patients be representative with regards to the population which they are dealing with? You don't know. The accommodation which is given by the pharmaceutical industry is actually a sort of house, an overbuilding, a sort of way of going. But when you are going to treat the patient, we have an individual situation. And everybody forget that. And we are very stupid people. Human is very stupid, actually. Because when we are doctors or treating a patient, then we have our white coat on. And then we try to do the best for this patient. But then we are taking off this white coat and going to the office and be a researcher. Then we forgot the way of thinking when we are treating the patient. What on earth is that? We are the same person. And we want to make sure that the treatments given have to be the best for the patient. Not all the patient, but the patient which you are in charge of at the moment. Of course, surgery have also a sort of evidence-based medicine. They haven't been performing so much trial as we used to have in pharmacy, but they have. And the claim for this kind of studies are not the same as for the pharmaceuticals. We accept that because they have the knife. So we have to accept that. We have other type of therapy, like manual therapy, where they're also treating the patient. They have changing the tablets with their hand and manipulate the organ in such a way that the treatment can be healed. We have physiotherapy, which is not the same, but have the same background. We have Chinese traditional medicine, and we we have to admit that these traditional Chinese medicine have a lot of experience, thousands of years. And of course, there is something there which might be also of interest for us. We cannot deny everything. We have to look into it and to see to which extent does that actually work. and what is the reason for its working. We have homeopathy, and we have others. So, when we shall prove evidence, should go into the field of evidence-based medicine, even it is on the level of a patient or a subgroup of patient or a huge group of patients, we need to perform that mainly based on what we call controlled clinical trial or randomized controlled clinical trial. So, I'm going now to look at what we are going to do in clinical pharmacy or treating patient on the single case or in a small schedule. What shall we do? What can we do? What can we do in order to get evidence which is on the same level as the one which we are feed up with so far? Well, we cannot perform large study for two reasons. We don't have the money and we don't have the patient. But we need to increase the design of the study. As I said, the big international studies which is performed in one and only design. It's a hopeless design called parallel group design, and then we need a lot of patients. A lot of patients. But we can be more sophisticated. We can use something that's over the head here. We have something on the top. We can use it. And we can use the fantasy. And I ask upon to you, all of you, when you shall perform studies or want to investigate, because a single case study is very much worldwide. It's very important to do that. Everybody say, no, it's only one patient. Yes, it's one patient. But how does this patient work? Because when we are working in a daily life, we are working with single patients. And why can't we get the single case studies and see what we obtain and report that to our colleagues? Isn't that evidence-based as well? I think it is. One possible design is several of them, which can be used in single case studies or small sample studies, is what they call multi-crossover models. It's a very powerful design. If you have small samples or laboratory studies, small samples in a university, in a hospital unit, for instance, or a small unit, lattice square designs is also powerful. If we want to look upon dose response or interactions between treatment, then we have the factorial design, or we have the more new one or more powerful one, which is called response surface pathway design. And if you want to perform studies which have an overview of the hospital to collect all your information, which all you and your colleagues actually obtain, you can perform what they call adaptive design study, which actually is an overview treatment in your hospital unit to see to which extent is the best way of going when you have a patient. So there's a lot of opportunity. The only thing is that you have to use your brain and you have to use your fantasy. You know, there is one thing which is very sad. There's several things which is sad, but one thing in research is that when we are boys, girls, young boys, then we have a fantasy. Look upon a young boy or a young girl. They can have fantasy. Speak with them and see you can stop their fantasy. But when we grow up, we are getting stiff. All over, except for one place where we shouldn't be stiff. But, you know, we get stiff. We are not allowed to use our fantasy anymore. We have to go in the same line as everybody. But as a researcher, you shall not go the same line. You shall go outside and take a look what's happened outside here. Can we do something outside? Is only the main road the main way of getting to the goal? No, it isn't. And there is more smarter way to do it. And as a researcher on the individual level, please do that instead of following the mainstream. We have challenges in over small units, in pharmacy and in all kind of treatment. We have to plan and perform. Single center studies with a powerful design, single case studies. We have to plan and perform control clinical trial related to small units, which means that it's a limited number of patients, but with a power equal to all the other clinical trial which is performed worldwide. And we have to investigate the effect and tolerability when we are combining pharmaceutical treatment with other treatment. Has somebody seen some papers of that, where we have pharmaceutical treatment combined with other kind of treatment? Which results do you obtain? Well, there is some papers, but there is not many. That is a huge field which is open and which is actually calling for researchers like you to perform such kind of study to see the interactions between the different kind of treatment which we will offer to the patients. And in order to do that, we have to increase our general knowledge in control clinical research methodology, because it's not in this group only. It's not only among clinical pharmacists which the knowledge is limited. Even in medicine field, different medicine field, they don't know. I've been performing and had speech and participation at ASCO in the cancer union, and I can see that their knowledge in performing clinical trial is not good. It's a lack of knowledge. And maybe it might be a good way to go for this group actually to increase our knowledge within this Congress by having pre-Congress courses, for instance, to increase the knowledge how to perform, how to optimize single-center studies, single-case studies, and small-scale studies. Which kind of treatment can we combine? Well, actually, we can combine a lot of. Pharmaceutical treatment can be combined with manual therapy, with surgery, with physiotherapy, maybe a part of, actually it's a part of Chinese medicine that can be combined with pharmacy. And we can combine also pharmaceutical with surgery and manual therapy. And we don't know today how will that work. Will that be a benefit for the patient, or are we not allowed to do it? Should we avoid it or not? Which kind of things is behind here? That is a question which is a challenge for us in this audience now, actually to go in front and try to do something with it. And I want to appreciate this Congress actually for being open enough to invite different kind of specialists in different fields of medicine treating patients. We have, during this Congress, there will be presentation of traditional Chinese medicine, there will be a presentation of manual therapy, and, of course, in pharmacy and even in surgery. So there is a lot of this is now invited in this Congress, and it might be a very good idea to look into the possibility of how this will merge together. Of course, surgical treatment can be also combined with different other treatment. Summary. First of all, increase the general knowledge in controlled clinical research methodology and statistics. That's an absolutely need. Perform and don't be afraid. Don't think that you are not so good researcher because you have only a case control, a case study. Perform such kind of studies because there is a lot of information in that. Perform small-scale studies with optimal design in order to show how this works in your unit. That's information. This information is needed not only for your colleagues, but in your unit, but also outside your unit. And please investigate the effect and tolerability which is obtained when you are combining different treatment from this basket of treatment. What will happen? Maybe we can make a better word for the patient in the future. Thank you.
Any questions for the presenter?
Thank you, Dr. Stig, for your comprehensive lecture. I am Dr. Nermeen Abu Saud, clinical pharmacy consultant from Prince Sultan Cardiac Center in Saudi Arabia. I have a comment and I want to share my experience related to the evidence-based application of evidence-based medicine to our practice. Now, you talked about the challenges to research or evidence-based, and there are many challenges at the patient bedside. One of them is the pharmacogenetics. You know that all the research and data came from certain countries. It's not applicable to our countries. Like I'm from Egypt, and I'm working in Saudi Arabia for more than 10 years. So the guidelines are not applicable because of we may see certain genes that may affect the treatment, like warfarin resistance, like metoprolol. There is resistance to metoprolol in our countries, in Saudi Arabia, and we started to make a pharmacogenetic makeup, and we're searching for the genetic makeup for the patients, and we find that not every research results can be applied. This is one of the problems. The other problem is certain medications are not available in our countries, in the Middle East. Maybe here there are many companies, we are not purchasing from them. So again, these evidence-based conclusions may not be applicable because this is the second barrier. The third barrier, as you mentioned, is the level of prescribers. Of course, the educational level of the prescriber affect the practice. And when we try as a clinical pharmacist to convince them about using the guidelines, and even the Pharmacy and Therapeutics Committee, I'm preparing guidelines for every disease in the hospital based on the evidence-based recommendations, but we find a resistance from physicians or consultants and even from the hospital director to follow these guidelines. So there is any practical solution to our problems in the Middle East? Can we make multi-central studies, or I don't know, can we collect data from other countries and taking into consideration the pharmacogenetics and the other problems? Thank you.
It's a good question. This is actually the results of what I'm criticizing, making a critical evaluation now, because when you got a new drugs, or if you even got it, that is performed in multi-center studies, not only multi-center studies, but international studies, including patients from all kinds of the world, and these are not necessarily, the results obtained is not necessarily related to you. That's why the clinical pharmacists have a huge work to do. Because first of all, the recommendation given for a drug or for a treatment, that's related to a huge population, and it's not necessary to fit you. If you shall say that you have to use X gram per kilo body weight 2 times per day, it might be that that's an average in the world, but it's not fitting your patient. That's why I said it is needed that you are performing studies on your patient in your country. If you speak about multi-center study, yes, but make multi-centers as multi-center or patient which is related to each other genetically or races or what it is. Of course, then you can moderate the results obtained, because pharmaceutical industry they say this is only the map. You have to use the map yourself. You have to finalize how narrow can you do. I mean, as a medical doctor, we give 1 tablet 3 times a day, and we think that all the patient is equal. They are not. But if you have a car and come to a fuel station, you are very upset with which kind of pump should I do. Huh? And then we are very needed to take the right pump. If you use diesel, or if you have fuel or whatever, and all these other things, then you are very accurate. But when we are treating the finest mechanism in the world, we are using 1 tablet 3 times a day. It's obvious that this is wrong, isn't it? So, yes, I apologize for being roof, but you are correct. But you need evidence-based medicine in your field, and that is up to you and your group to perform it. I'm sorry to hear that not all medicine is available. I know, because I've worked in Asian country as a doctor myself, and I know what's available, and I know the difference between the patient. Thank you very much.
Maybe just I comment also on that nice question. I really appreciate this question. This is a very important issue. And this is basically well documented that there is ethnic background that are actually affecting, the gene structure that actually affect the outcome of the therapy through the SEP system and the P450. All of these things are well documented. However, remember that all of these clinical trials are done by what? By drug companies. And this is basically a profit-based sort of business. So they are more interested to have these trials done and finished, start and finish early and as adequate as possible. That's why all the trials are basically for new drugs are from North America, Europe, maybe Japan sometime. They don't care about this type of ethnic backgrounds. And you are right, because it is proven that warfarin therapy is really affected by the genetic structure, and also a loss of neurological diseases are treated by phenytoin or other drugs that are affected. Cancer therapy is a big thing right now. What you can do, simply this is documented right now. And I think, again, as Dr. said, this is your responsibility. Science is there. Pharmacogenomics is evolving and improving and increasing science right now. It is well documented, lots of literature in the field right now. What you need to do is just simply to repeat these studies that are done somewhere else in your local, situation. And it's easy, by the way. It needs some funding. You can just have some sort of political power there on drug company there, or maybe you just initiate studies. And I know lots of hospitals in your area, in the Middle East, in the Far East, they are starting these type of studies. This is evidence-based medicine, but this is a must. This is basically a must. This will actually improve your therapy for the patient and the patient outcome, and also our knowledge and education about medication. Okay? Thank you. right, so welcome back. I think we spent more than 6.5 minutes. I think it was 11.225, right? But I think we are here, so this is great. I'm just going to continue in my thread of thoughts about clinical pharmacy. We talked in the morning about the profession, and we talked about changing the profession. There is evolution in the profession. And then our job is to try to respond to these factors and begin to modify our profession and build new models to achieve our goals as a well-defined, successful profession. And I believe that the route for that is through education. So when we have any factors changing the profession, different things that can modify our goal or our direction, we have to respond to that. And we have opportunities. And we had also lots of missed opportunities. So I think the best way for us is to try to understand what's happening around us and then begin to modify our route. And I still believe that the best way to modify the route is to start by education. When we have new direction, new models, we have to prepare our graduates or our pharmacists to teach them the new tools, give them the tools, and get them more up-to-date and more advanced education to be able to perform and do the change. We are not going to do the change ourselves. We are getting old enough to not to change things ourselves, but we can influence the change process. And again, this is for the third time I say that, it has to be through education. And the good news is that that's exactly what's happening right now. As I discussed this in the morning, I said that there are lots of things happening to our profession. It's evolving, and now our job is to respond to these factors and these changes and take advantage and seize the moment and the opportunity. We don't want to lose another opportunity as we lost before. We lost lots of opportunities for our profession. You know, let me just give one name, the opportunity for pharmacists to work in the field of radioactive materials and build a profession around these things. We lost it, but we lost other things also. Now we cannot afford to lose opportunities. Now we have opportunity. I'm going to explain to you. I think I mentioned something about that, but there are opportunities that are there, and it is into our side. And these opportunities actually are there for us to take it. We have to take it. How can we do that? Again, for the first time, through education. We have to prepare our pharmacists to be competent enough, to be professional enough, to be educated enough to perform in these new models. Otherwise, we are offering nothing, and these opportunities will be lost. So I'm going to focus today about what we are going to do to respond to these changes and what this affected our education or pharmacy education. I received my BS back from Egypt, 1976. So at that time, I received some type of education, which is totally different than what I'm doing right now. I'm now teaching, so I'm teaching different model, teaching different materials, different directions, different objectives, with different people, different students. It's different story, different game. So we need to understand the factors here and begin to adjust based on that. So I'm going to talk about the change, that trends in the pharmacy education that happened accordingly or based on the change in the society, change in the health field, the change in the way we treat our patients. And how can we adopt to this change and seize opportunity? We don't want to lose opportunities anymore. And then how can we move to make these opportunities or these moves a real successful one, building on success and compliance and assessment and proving evidence-based professional improvement. We have to show that we already improved our education, and this way we can say, hey, this is what we're going to do as a profession. So let's just think about it, and I hope that you can interact with me so you can give me your thoughts here. This is based on my experience from where I grew up, from where I worked. I worked in Saudi Arabia, by the way, sometime. I worked also, I had exposure to different schools in East Europe, even in Far East, and now in Canada, and then in the US. So I had some sort of exposure to different models of education, and I was following what's happening in the field of pharmacy and what's happening in the education process. And as you know, we started from the model, which is basically the simple pharmaceutical sciences model, where we teach students how to prepare the medications, how to analyze the drugs, how to make the analysis, and then how to sell these drugs. But nothing beyond that. Now the model is totally different based on different factors that happen here. We have healthcare challenges. There are changes here. I'm going to talk about this US system here, but I think we can, maybe with your help, we can just sort of think about it in different models. I think in Europe right now, the model is beginning to evolve, and I can see even in the Middle East and the Far East, and I think sometime in Africa, they are starting to think about, especially in Kenya, starting to build some new models for pharmacy practice based on the healthcare challenges. So we have the healthcare challenges, number one. We have different students, different mentality, new generation. We have the millennial generation. These guys are totally different than what I was when I was a student, and what was my late dad when he was a student. Totally different. The way they receive information, the way they digest information, the way they even present this information is totally different. And also accountability is a factor here, because nothing is going to be happening if we don't prove that we are accountable to what we are doing. We have to show evidence-based education. So evidence-based is everything. In professional evidence-based and also in education, we have to show them that the outcome is well documented, the outcome is proved, and we show them that we did that. This is through the assessment process. These are requirements that are now, pushing us to change the way we do our business here. So let me start with the healthcare challenges. And basically, at least in the US, it starts by lots of factors demanding lots of healthcare workers. And this demand based on new different factors happening here in the society here. America is aging. Lots of population now are above 55, 60. The baby boomers, now they are now 65, 70 almost. And these baby boomers now began to grow, and now they are aging right now. And this is a big population. So, and due to the increased, accessibility to medication, to therapy, to advanced care, now patients don't die. They stay longer time. So we have a real big number of population are aging. And those aging, personnel, we are really eager, or we are responsible to treat them. So we have to offer them healthcare, and this is big demand right now. This is one challenge. Maybe in the different countries, I think it's not that factor there like aging. I think the Far East is basically young population. We have lots of different, population is growing, and all of them are youth. So maybe 70% of the population maybe are youth. So this is another factor. So we have to look at it in different dimensions. We have to treat the aging population in the US, but also maybe in other parts of the world, we have to worry about, the younger population. But both of them has to be taken care of. Both of them has to be treated, and that's why there is lots of demand for healthcare professionals right now. Also, you see every day, every other day, we have a new drug, new medication, especially for the new technology, biotechnology drugs right now for treatment of cancer and immune disease. And all of these factors here are increasing the pressure on us, because there is lots of drugs every day, every week. We have new drugs, registered, approved into the market. So I remember when I was in my pharmacy, 30 years ago, we had couple of hundreds of drugs. Now I think they are doubled or tripled. So we have lots of drugs right now, lots of medications. And now we began to have lots of knowledge about the effect of this medication, like the pharmacogenomics factors here. So the same drug, warfarin, that we used to treat patients with 30 or 40 years ago is different, because we discovered that the same drug can affect patient A, in a different way than patient B, than patient C. And these factors are very important because this is life-threatening conditions. So now we have lots of information about medication, therapy, number of medications, number of reported adverse effects, toxic effects. It's huge forest, or I call it jungle of information that's around us. And the best profession that's really well suited to deal with this change are the pharmacists. So now there is big demand for pharmacists to take care of that. This is an opportunity. We have to step in and take advantage of that. The last thing is basically the dynamics of the healthcare. At least in the US here, the new directions right now from the Institute of Medicine is simply to allow all the patients, no exception, access to healthcare. No exception. And access is a key here. So when you say access, mean that this patient can have access to treatment maybe at home, maybe in the clinic, now in the pharmacy, in the hospital. Access should not be just only in the hospital or in the clinic. It has to be even in different areas. You can have home health care, across the board. And this is basically an opportunity because now we have demand for professionals to step in and offer that service. Till now, we are losing because nurses took over. Let's put it this way, in the US at least. They took over the primary care, professional work. So now they could push politically and maybe scientifically, and they graduated lots of nurses. They developed new programs, PhD program, nursing practitioners, clinical nurses, you name it. And they proved that they are efficient enough. And now they are licensed to treat. So they have now clinics, primary care clinics, run by a nurse who take care of the patient as a primary care professional. Pharmacists lost that opportunity. And I cannot give any excuse to the APHA, ASHP, all of these guys who are meeting every day and making parties and celebrations, and they are losing this opportunity. This is not an excuse. This is a crime for our profession. That's why we have to push hard and say, this is our right now to take this opportunity. Now there is lots of opportunities now for ambulatory care, for patient care, for lots of things that we can offer. We just need to educate our pharmacists to do it. And we have to produce well-educated pharmacists trained to offer that job. If we offer a wishy-washy pharmacy or pharmacist to offer this job, doesn't know what he or what she is doing, no way, you are going to lose again. We have to prove that we have a product that can deliver, and these deliverables are successful deliverables. So we have to look at education again, either by continuing education for our existing pharmacists through lots of all of these workshops, all of these certificates, all of this, even new licensing. And also for our graduates, we have to change the way we teach our students to make sure that they are ready to face these challenges. It's a perfect storm. Basically, we have lots of factors getting together and create an overwhelming pressure on us. And this is a storm that we have to ride. We have to be ahead of that storm. We have to really push hard to be successful and get out of that storm successfully. Otherwise, we are going to be left in the dust as we used to have all the time. We have to take that opportunity. This is very important. How? Through education. So what happened? And I'm just mentioning some examples here in the US, and I know that some examples also in Europe, I think in US, in Great Britain, in Germany now, even Germany that have classic pharmacy education, classic pharmacy practice, now they are moving into new models of pharmacy practice. Now we have increased responsibility of pharmacists to deal with chronic diseases, medication management, disease management. Now we have medication therapy management. These are responsibilities that are now taken care of by pharmacists. Also, we have drug benefit plan management. We have pharmacists who are specialized to make sure that you can modify the medication plan or the benefit plan, the insurance plan, to make sure that it can fit what we need to do for our profession. Medication therapy management, as I said again, this is a new star in our profession right now. And I used I built actually a center for medication therapy management when I was in Virginia. It's a very successful story. You have clinical pharmacists, taking care of patients with chronic diseases. They have an appointment with him or with her. He or she spend maybe 30 minutes to 1 hour consultation service, going over their medication, going over their lab tests, communication with their physician, with the nurse, going over all of these factors for the therapy, and then ending up formulating, we call it medication therapy plan or therapy plan. A well-detailed plan that's communicated with the patient. Even we give them, hard copy of their medication plan, and we already do some sort of consultation and go over these points with them. Then the patient come maybe after 2 to 3 weeks for follow-up, and then another follow-up, and then another follow-up. i.e., we are responsible for the medication management for this patient with hypercholesterolemia, with diabetes, with hypertension, whatever. This is an opportunity. This is a profession. This is a new profession, by the way. And by the way, we get paid for this consultation. So insurance pays us for the time we spend. Every 30 minutes, I think we are paid $150. This is really because unless you get paid for your service, it doesn't exist. If you offer service for free, it's not recognized. But if you are paid, this means, especially if you are paid by the insurance, you are recognized. This is a success story that it has to be spread all over the place. It has to be spread where you practice. It has to be spread all over the world. And I think there are some models like that in different ways. I think I heard about the UK that they have a different model. They have a prescriber pharmacist, a pharmacist who is specialized with a special education to offer specialized service to take care of patients with specific diseases. We don't have wishy-washy pharmacists who just treat everything. Nobody will know everything. But specialized service is very important. You have a pharmacist who is offering infectious disease specialized. I'm going through even certification now for infectious disease certification for outpatient clinic because I would love to do something like that. I did lots of my work in the hospital with antimicrobial stewardship things, but I want to test it to see how can we offer infectious disease, service in the community at the same time for antimicrobial therapy. It's cool, it's beautiful, it's exciting. Also, pharmacists now are moving more into research and translational research, clinical research, and might add also practice-based research. You talk about outcome research, where you document your service. If I have an intervention for patient with asthma, I need to document my service by, i.e., pre and post intervention. I measure the factors, the clinical factors, the economic factors, all of the other factors, pre and post, and then I can document that. This is what we call practice-based research. This is huge right now. We need to work on these things. We need to prove that we are offering service and document it, and this is very important. Then I go to the last step, which is basically team-based practice, and this is the new directions right now. Interprofessional service is the key right now. Nobody is working the solo there. Nobody is working just to take care of the patient. You don't have that physician who just say, hey, I'm God, I can take care of this patient. Let me make sure that he is going to be treated. If he dies, okay, this is God well, I don't care. Nobody will ask me about these things. No, my friend, it's not going to happen anymore. You are working within a team, and each part of the team, each component of the team has some responsibilities. You are working with the physical therapy, you're working with the pulmonary therapist, you're working with the clinical pharmacist, with the nurse, with everybody, specialized service. Each one of the team will work together to have a therapy plan and monitor the patient. Because the key thing here is to make sure that we give the patient the best therapy possible with minimal adverse effect and the least expensive one. This is very important. So the team-based sort of practice is the key right now. So part of that team is a pharmacist work. So we have to identify our tools, our talents, our intervention, our key points, and be good at this, and then we can be part of that team. And again, this is also another evolution of our profession that increase our exposure as professionals who are offering therapy and treatment for the patients, not just only selling medications. So The health challenges are there. The second part is, so now we are changing all of our educational models to produce pharmacists who can follow these directions and actually be active in this new model. So now we are in our curriculum, where I'm now taking care of the education in Chapman University. We are building a new curriculum, which is basically integrated. It's evidence-based. It's basically, active learning process. We get them into cases. We get them to learn how to know about the drug, but also how to use the drug, and then know how to monitor the drug therapy. We just get them into that field, and we give them lots of training about, medication therapy management, about immunization, about, ambulatory care, diabetic care. All of these things are part of their education. We are trying to adopt our training, our education, to fill the needs. And this is going to be the success story. The second way we have to change our education model is to respond to our new customers. We have new customers here. We have the millennia guys. They are different. They are totally different. They spend at least 70% of their time on the computer or the cell phone or iPad or iPhone. They are computer experts. They are technology savvy. They receive things through the internet, nothing about books anymore, or literature or lecture. All of the things are garbage for them. They just want to get things through the iPad, iPod, broadcast, all of the things. This is a new way of thinking. And they don't believe in memorizing. They have to think. They have to find the solution themselves. They are smart. So they are different. So these customers, we have to change ourselves. It's difficult. We are used to do something classic for years, for hundreds of years. Now we have to change. There is no other choice. And that's what we are doing. So we change. I'm talking from my experience because I'm responsible for the academic program in the school. So we had to change the curriculum to fit with I think this is College of Advanced Pharmacy Education from the AACP. They have criteria. You might go and check there. They have criteria to help us to build new curriculums that will fit the needs of our students. And this is very important. It's active-based, active learning. It's more into teamwork, interprofessional education. This is very important. And also the way we deliver this education is totally different. I remember I have lots of my colleagues. You talk about guys who have been teaching for the last 10 to 30 years, and then they ask them to do something different. They are crazy. They just say, hey, are you nuts? I was doing that for the last 30 years, and I'm a successful teacher. I do a good job, and I enjoy it. Now you want me to do it in a different way. You are getting me out of my comfort zone, and they are right. But sorry, we have to do it. Because we can't do the same thing that we were doing for the last 30 years. We have to change. Change is there. We have to adapt to what's happening here. So, for the curriculum change, we follow the CAP outcome, where you have collaborative work, interprofessional education. I have 3 courses right now for IPE, interprofessional education, where we meet with physical therapy, with physician assistants, with nursing, with medicine. And we have a course that's taught for all of them at the same time. And this course is basically case-based. In fact, next week I'm going to introduce to them a new program, a game, a video game. This is typical millennial thing. We're not going teaching. We're going to have a game for students to play together. It's a video game, and they play together. But this game is about a case, about a patient. So the physician, assistant will take it from that point, and then the nurse will take it from the other point, and then the physician will take it from that point, and then the pharmacist will talk about therapy and therapeutics, and then the physical therapy will talk about, physical therapy issues. The same case, but it has to be tackled as a team, and this is exactly what happens in the real world. You have a patient, and you have a team working with them. We teach them, we train them how to do these things in a game format, so they will love it. I remember that we had some session, students loved it. It's just beautiful for them. It's just each one has a computer, we have a game, and then the game is in the cloud, so they share the same game, the same patient, and say, hey, do that, add this drug, remove this drug, do this diagnosis, do these things, in the same case, and they are connected to each other. They love it. This is the way they do it in the game, I think Game of War, something like that, which is famous game, the same model. They love it. It's education by playing through games. And they love the things. And this is typical millennial, approach. I'm lucky because I have lots of time to talk, so they gave me permission to talk double time because there's not enough speakers. Lots of the speakers already could not make it to the conference here. So I was confirmed that I can talk more than what I should do. So I'm feeling comfortable now. So bear with me. So we have interprofessional education. Leadership, we have to have leaders. We don't have to have those guys, bookworms who know how to memorize and get an A, and they are really bad personnel. They are not really good, leaders. They just know how to memorize and get an A. We don't like these guys. We prefer someone who is active, proactive, thinker and leader. This is very important. Personality is important. And then we have to have a student or a pharmacist who know how to deal with technology. You have a guy who is 61 years old like me, who I was lucky because I like technology. So I trained myself with that. But I know that it's difficult for us to go and learn new technology, new programs, new programming, new things. It's difficult, but we have to. Because we have to train our students or our future pharmacists how to use technology, because this is the future. You are going to have technology everywhere. In fact, there are lots of iPads right now, or maybe iPhones right now, and maybe small handheld handheld computers that you can use it for following the heart rate and blood pressure and maybe some diagnostic things. Lots of health-related issues are on these apps. Lots of apps. I just purchased an app yesterday that to try to do modeling for treatment. Everything is through apps, through computer programs, and in fact, not even computer. Now the iPad and the iPhone and the Android is the key right now. So we have to learn these things. We have to learn how to do the things, because this is the future right now. We prepare our students, but also we prepare ourselves for that. Now, also, we have to change the way we teach our students. What's the classic teacher style that we used to have? The same thing that I'm doing right now. I'm standing here, tell you what I know. You are sitting there, listening to what I say, maybe are interested or not. Some of you guys maybe fell to sleep. Some guys will say, oh, what is this guy doing here? I don't care. I don't want to listen to these things. This is classic lecture. It doesn't happen anymore. We are still doing that in conferences, but I wish sometime in the future we can have some sort of interactive model there. Now in the classroom, there is nothing like that. We have to change the way we teach our students. The way of active learning pedagogy is very important right now. And now what we call it pedagogy, I call it andragogy, by the way, because we are dealing with older. Pedagogy is from pediatrics. So this was developed for children to learn how to learn. But now we are dealing with grown-up adults, so I can call it andragogy. I'm going to change that. And we call it flipping the classroom. So changing the classroom dynamics. So other than teaching, and you guys are receiving, no, I'm not going to teach anybody anymore. I'm going to sit there and allow you to learn. So it's not teaching anymore. It is learning. And this is far more comfortable, far more powerful to get the learning process other than the teaching. This is what Einstein did a couple of decades ago. He just said it, and this is very brilliant. Nobody knew that, but this is really brilliant, other than inventing all of these beautiful things. He said, I never teach my pupils or my students at that time. I only attempt to provide conditions in which they can learn, period. And that's exactly the essence of what we're doing right now. We're not teaching them anymore. We are going to get them into the condition and the mindset and the way and guide them to learn how to learn. It's a lifelong learning process, journey, and we are going to help them to get into that stage. So based on Bloom's taxonomy, all of us received education at that level, which is memorizing, remembering. You remember when you was in the classroom? I used to hate my A student friend, who was the A of the class. I wasn't, really the top of the class. I was maybe number 4 or 5. But I hated this guy because he just had a memory that's really super memory. This guy could just give me the textbook and say, hey, Ayman, can you just please, listen to me? And he just typically says what's happening in the textbook, page by page. If he said me, change the page, please. Okay, thank you, my friend. He is just a genius in memorizing. Is he a smart person? No. He just has one talent, which is memorizing. But the problem is that our education system at that time was favoring those memorizing things. So you have all of those guys who got A or straight A's are the ones who are really able to memorize things and have photographic memory so they can get good A's. But you can have lots of smart guys, innovative guys, and it was proven when they graduate. I could see that. I made some sort of study to see what's happening to our graduates, those who got A's and really top of the class, where they are right now, what they are doing. And those who did not get A's, but they are okay, maybe B's or A minus, whatever, but they are not really top A or top of the class. They did better. They are innovators. They built companies. They built even schools. Some of them actually are deans of schools right now. I remember a friend of mine who was one of the, I think he's the richest or the most successful pharmacist in Egypt right now. He was failing the courses. He was a C or D student, but he is the most successful pharmacist. And this is really amazing. It tells you the story here that to be successful in your profession, you don't have to be with a memorizing. You just need to have an innovative leadership, open-minded, and an entrepreneur type of thing, eager to learn and know how to learn, not just memorize. So now we are moving from the memorizing to the understanding. No, we are getting even more. We are now teaching students how to analyze the situation, how to evaluate the condition, and how to create and develop something new, i.e. treatment plan. This is what we want them to do. We don't care about memorizing. I'm not going to have a patient and say, oh, you have hypertension. Oh, hypertension is a disease that's caused by 1, and this is what's happening, and this is the treatment. It's not going to go anywhere. The patient needs from you a thinking process. Oh, you have hypertension. What about the heart rate? What about the blood pressure? What the other drugs there? If there is something that can influence this blood pressure, if there is some medication he's receiving, if there is some food thing that can affect that, what about the salt? What about the levels? And then get things together and then say, hey, you need to do these things. These are the plan, or this is the plan that you want to follow to get really good control of hypertension, not by just telling them everything about the hypertension in the textbook. So now we are shifting more into analyzing, evaluating, and creating, which is more important thing. Do you remember a movie called Rainy Man? Do you remember that? Tom Cruise with Dustin Hoffman? Yeah. And this guy, he had autism, right? A type of autism that is basically, he is genius in memorizing. Only memorizing. So I remember that, this is a beautiful scene. I remember that when Tom Cruise gave him a phone book, he literally memorized the phone book. So he could go over it, and he could tell him, page number 5, oh, these are the phone numbers, and these are the names. Page number 7, these are the phone numbers. That's wonderful. So this guy is an A student. This guy is brilliant. But when he gave him a phone and told him, can you dial the number, please, for me? He couldn't. He couldn't. Because he knew how to memorize, but he didn't know how to use this knowledge to do something, to evaluate the situation, to create something, to do something. So we are looking for guys who know how to do things, not to memorize things. This is exactly what we are going for right now. So now the dynamics in the classroom are different. We are dealing in a classroom in a different way. We used to have the lecturer standing here, and the students are there, and then I give them all the information, they receive it, go home, memorize it, do the homework, come to the exam, vomit the answers, and then forget the whole thing. That was the classroom, right? That was the way I was raised, the way I received my education. It's different right now. Now we have the lecturer standing there, doing nothing. All the information was given to them a priori. And in fact, not all the information to memorize, all the directions, how to dig in, how to find information. Go and dig in and find articles about the treatment of hypertension. By the way, there are guidelines, find it. What are the recent guidelines? By the way, there is a video there about the treatment of hypertension. And by the way, this is my lecture. So I used to record my lecture. This is what I'm going to tell you about what the knowledge there. Here it is. Listen to it. Dig in, find information. Read articles, read the guidelines, learn about it. Google, find everything. So you come to the lecture with the knowledge already digged in and found, and you got it, and you spend the time and the luxury and the knowledge to find it yourself. So the knowledge that you find it yourself is far more effective than the knowledge that you get it from somebody else. When you dig in and find something, it is there. It doesn't go away because you spend some time and effort to find it. Now we come to the classroom. I don't lecture you anymore. We get together in groups and we go over cases. We go over discussion. We go over important points. We go over, play a game. We go over a game, and we try to interact. And within that process of interaction, the knowledge that I gain through finding it gets there. When I get a case and begin to think about it, and then my friends say an idea, I get an idea, and then an opinion, another opinion, and then I give them my opinion, and we formulate something together. This is education. This is active learning. That's why we call it active learning. So the lecturer or the professor is not the sage on the stage as you saw. It's he or she is simply the guide on the side. That's the exact expression that you used to have here, just to guide. You are not a professor anymore. You are not a teacher anymore. You are a guide. You are a preceptor. You oversee the activity. You try to roll that activity and make it more active. So in the traditional education, you have the teacher there and everything around him. He is the professor, so he is the one you have to get afraid of because he's going to give you an exam. Information he tells you or she tells you is there for the exam. And he's threatening or she's threatening you because this is going to come in the exam. This is a question exam, information for the question exam. This is a really threatening environment. It's not anymore. It's knowledge-based, just the knowledge to memorize it. One size fits all. I give you the information. You are going to adopt it. You are going to understand it. You are not going to understand it. You're not going to even think about it. You forget it. You are sleepy. I don't care. It's just I'm telling you everything. One size fits all because we have different levels of education, different level of thinking and receiving information, different way of absorbing information. So we have to consider that. In the traditional way, we just one size fits all. These are the information. Memorize it. I don't care. That's it. And what we call it here is a passive learning. In the new model here, we have education is student-centered. You are going to dig in and get the information. You are going to teach yourself how to do the things. You are going to find the new knowledge yourself. It's a performance-based. I'm going to test you on your performance, the way you find the thing, the way you perform, not the amount of knowledge that you can memorize for me. It's a differentiated instruction. It's because I deal with each student in a different way, based on his or her way of thinking, if her way, mental status even. Some student may be depressed today. I have to deal with him or her at that level. Some student is just active. I just deal with him or her at that level. It depends. It's personalized education, as I call it. And it's active learning. I'm going to go over that. So just simply we have everything before the classroom. And then when we come to the classroom, we come and do role play. We have practice-based. We have cases. We have different ways of assessing that. There are challenges. I'm not going to go over that, but it's very important. The most important challenge that the professors themselves, they feel threatened because this is a new thing. You are asking them to redesign the course. You have lots of faculty work, and they are going to be very resilient. And you have to give them professional support. You have to have a system around them to help them to do all of this panopto recording. They have to learn how to do this technology. You have to have some support system. You don't leave them and say, hey, fill up your classroom. You have to give them support. And what's more important, that you have to convince the students to buy into the process, because these guys came from high schools or maybe college with the old way of learning. Now you are showing them a new approach. It's a transition for them. You have to get them into that. You don't just push for them. You just get them gradually and get them to learn this process. So this is the third part here. After you do all of these things, you have to show accountability. You have to show what you are doing is right, that the results are positive or negative by doing assessment. And this is very important, that whatever you will do, you have to assess it. You have to have continuous assessment. You set the goal and the approach, and then you gather the evidence by assessment, maybe formative or summative assessment. But what's more important is that the data that you get from this assessment process, you don't throw it in the garbage can. Or don't say, oh yeah, I had all of these evaluations, and I got at least 5,000 evaluations on my desk here or on my desktop, and we did a great job in the assessment. No, you have to use this data and show that you analyze this data. You have spent lots of time analyzing this data and getting lessons out of this data and begin to change direction based on what you found in the outcome here. And that's what we call it, close the assessment loop. You have to use the data to begin to change your goals, change your approach, then do assessment again, and then analyze the data. If there is change, you change and continuously. This is what we call it, closing the loop, which is very important factor in the education process here. Let me give you this is my personal vision and reflection to make sure that you can, if you want to take care of an academic program, these are the steps. I tested that, I did that sometime, I was successful sometime, failed sometime, but simply you have to set the goals and develop short and long-term plans. You have to have the faculty involved in that. And then you have to maintain viable, build a competitive academic program that meets, at least here in the US, the ACPE. I think there are some sort of guidelines in different countries. I think ACPE is now expanding to just oversee lots of other programs, and I think that they are successful in helping other programs to educate pharmacists. And then you have to make sure that you take care of your faculty through faculty development, improve their talent, improve their capability, and also set a culture of partnership. You have to build a team, and each member of the team has to be responsible for this type of things. This is how we can get ahead of the storm, and this is how can I say thank you, guys. So, if there's any question, please.
Thank you, Dr. Ayman. I agreed with all what you said. I want to say that I don't get the priority or the chance to go to America or Canada or to learn about clinical pharmacy, but I lived all what you said, I practiced it already. First, I want to talk about documentation, which is very important for any clinical pharmacist want to establish a service in a hospital. Documentation is very important because I worked as a clinical pharmacist in about 7 subspecialties in 7 different countries in Egypt and Saudi Arabia. To convince the community, starting from the hospital director to the residents, consultants, pharmacists, we have to document everything that we are doing. All my interventions and recommendations with the patient file, age, name, and everything I document. And finally, I got to this, my poster, which will be presented in this conference. It is that comparison between the different types of medication errors in 4 different hospitals in 2 different countries in the Middle East. The study period was from 2005 to 2015. In each hospital, because I worked in oncology, then I went to tertiary hospital, internal medicine department, then to pediatric hospital, then finally to cardiology center. So I'm comparing the incidence of medication errors or my recommendations in these 4 different centers through my data that I collected over a period of 10 years through my documentation. I selected 100 files from each hospital, and to get this data, I have to work for 10 years. Without this documentation, I cannot I established the service in these different hospitals, and without nobody in 2005, nobody told me that I have to document, but I felt that I have to document. Otherwise, if I'm not documenting, I'm doing nothing. Nobody knows what I'm doing. What's my job description in these countries, in these hospitals at this moment? The other thing is a successful story. Yes, we have to convince the community with a successful story. In 2012, because the quality requirements in the hospitals, they started to make something like improvement projects for the pharmacy department. And when to make these improvement projects, they asked the pharmacists to participate. So I participated in making an improvement project for the pharmacy through also detecting the medication errors or drug-related problems, because this is my PhD thesis. And finally, I found that about 60% of errors occurring during prescribing, 58% occurring during administration by nurses, and 55% occurring during monitoring. And I made an improvement project of 5 steps. The first step is implementing the clinical pharmacy services. The second step is to educate the staff about the most common errors that I detected. Then in our hospital, there was no IV admixture unit. So I recommend that we have to have an IV admixture unit to decrease the incidence of medication error in administration. Then establish a drug information center, and the pharmacist has a role in the drug information center. So this was one of our recommendations. Over a period of 6 months, I decreased the incidence of medication errors from about 60% to 10%, which is the benchmark in the international guidelines. So a successful story should be published, and everybody in the hospital should know about it. It is a successful story, so everybody should know. The other thing is using the technology. In 2005, I was working as a clinical pharmacist. Every day I'm seeing different cases, different drugs, different adverse drug reactions, and I want to collect cases for my PhD. But I cannot memorize all the side effects, all the dose adjustment in renal and hepatic impairment, all the interactions. I cannot memorize. So at this 2005, I got this ball, the pocket computer, before the now Micromedex and Medscape and Lexi, everything is in our mobiles. But before mobiles in 2005, I brought this Lexi pocket computer. It was a pocket computer. And all the time, all my rounds, I always checking. I cannot memorize millions doses of different drugs. I cannot memorize. Some needs dose adjustment in kidney, some need dose adjustment in hepatic. There is interactions, there is contraindication, relative adverse drug reaction. So I have to have this technology. And I started using this technology from 2005. And it was very successful. By this technology, I worked in many other different subspecialties. I'm so sorry, I was so long. The final comment.
Can you have a speech and present all your things instead?
Huh?
Can you have your own speech and present that in a separate thing? Because
We just have her finish.
I have a presentation.
wait for your presentation.
Finish the
I
last
have a
point.
presentation about my booster by chance in my flash memory. So if you want, I don't mind. The other thing is that I worked as a lecturer of clinical pharmacy, and I'm now a clinical pharmacy preceptor and assistant professor of clinical pharmacy collaborative, because I'm in the hospital in Qasim University. And I experienced using the team-based learning in our students. Actually, there are many challenges and many barriers to use this system. I'm talking about my experience. The first thing is our students are not familiar with this system. It's only, we started to apply it in the college, in the university level. So primary and secondary level, they don't know about anything about this. So they cannot, as you mentioned, they cannot understand. The other problem is we are giving the students one chapter from applied therapeutics to read and understand and summarize in 1 week and come for the exam, and the exam with marks. And we give them 10, 20 cases to actually, pathophysiologically, يعني, they cannot understand, they cannot, it's very, very problem. Yeah. The other problem is it's decreased the level of the educator.
Yes,
because
I
it's not...
Having your own presentation.
Let me just...
I need to know if you have any questions.
I think let me just have some comments here. I think I appreciate what you are doing here. You know, I really applaud your efforts, because I can see that you are sincere in your profession and you are struggling. Because this is we struggle all the time. You know, I know that. But just to go over these things, don't feel upset because you are not here in the US or in Canada because you are not learning clinical pharmacy. Clinical pharmacy can be anywhere. You don't have to be here physically to learn it. You can have it there by just having education, continuing education, access to these things. And there is lots of accessibility right now. And I know that from Saudi Arabia, I know that because I know that the ACP is working hard to push for these things right now. Number one. Number two, for the technology things, yes, you are right. I had the same experience. It was 1997. I got my first PDA, and I was always going through the same issue, and this is the best solution. Now, we don't have a problem like that. Every there in the PDA, you don't have to worry about carrying all of textbooks and memorizing things anymore. Now, for the experience with the team base, this is culture change. And to get it, you have to build it. And as I said here in my first moment in my speech here, everything could start from education. You have to educate your new graduates, your pharmacists, how to work as a team, your even physician students, your medical students, nursing students. Everybody has to learn this culture. You have to learn working together. In the classrooms where we grew up, you have each student alone. But in the US here, they work in groups. This is a good thing about education. They teach them how to work in a group, in a team. And this is now is going to be reflected in the higher education, that we as professionals will work in a team. Once you have this learning module from the education, it will jump into the profession. So you have graduates who know how to work together, understand the advantage of a teamwork-based things. Okay, thank you again.
So, thank you very much.
Very good morning to all of you. I think Professor Iman has provided a good stage for me, when he talked about pharmacy education for the 21st century. I come from a practice background, working in a hospital, and certainly, pharmacy education has continued to evolve. There are many challenges in healthcare, as mentioned by you. And that includes, obviously, demographic changes, healthcare models, new drugs that come into the market. And therefore, how pharmacists practice, in the practice area makes a great difference, and that is actually a continuum of education. So my presentation today is about factors that influence the effectiveness of pre-registration pharmacist training in a tertiary hospital, and that's my hospital in Singapore General Hospital, the views of preceptors and trainees. The outline of my presentation will cover the background of pre-registration training in Singapore and in my hospital, the objectives of my study, the methods, results and discussion, conclusion and future work. As a background, pre-registration training prior to licensure is an important continuum of education from classroom teaching as an undergraduate to real-world practice. Therefore, effective training is critical in order to prepare pharmacy graduates ready for independent professional practice. This is the first study of its kind conducted in my hospital, the Singapore General, and also locally in Singapore, to seek opinions from stakeholder groups, the graduate trainees, and our preceptors to better understand the key issues of pre-registration training. As a form of background, at a national level, 1 year pre-registration training is a regulatory requirement under our Pharmacist Registration Act 2007. Typically, this is conducted 1 year post-graduation. In 1997, we had 3 months in-course and post-course 9 months of the training. And going forward in 2017, the pre-registration training will be done 6 months in-course during their undergraduate days and 6 months post-course. With this as a backdrop, you can see that we will need to adjust our training. In any case, the new training modules from 2017 onwards will involve 4 rotations in 1 year, so that would mean 3 months, and 2 of which are compulsory, and that is in acute care as in a hospital, community care as in, you call them community pharmacy or retail pharmacy environment, and then the other non-core components can be indirect patient care such as regulatory affairs, manufacturing, the pharmaceutical industry, or primary care, and another posting would be in ambulatory care or specialty centers. As mentioned earlier, the 2 rotations of 3 months will be done in course and 2 rotations post-course. So in preparation for this, my hospital, which had previously been providing, if you can see under previous model, the 9 months training post-course, we have our trainees rotate through different sections of the hospital, all right, and because of this new program, we plan to then revamp our training model, and we will be providing 2 rotations, one in acute care and the other in ambulatory care, both 3 months each. So the objectives of my study, right, is to examine preceptors and trainees' perceptions on the factors that influence the effectiveness of pre-registration training, and then to identify areas for improvement and to develop the strategies to enhance our current training program and learning experiences for our trainees. Methods. We started off with 2 focus groups, one each with preceptor and the other with the trainees. They were held separately to gather opinions and elicit elements for our survey questionnaire. The preceptors and trainees of the 2014 cohort were randomly selected, and for optimal discussion, the target was to have 6 to 8 of them for each session. The elements that were elicited from the groups were then grouped together under the factors training program, preceptor-training relationship, trainee-related factor, preceptor-related factor, and environmental-related factor. The survey questionnaire was then formulated, incorporating the elements from the focus groups, literature, and the author's personal experiences. It comprises then 50 items on an 11-point scale, and this questionnaire was then distributed to all preceptors and the trainees in the department. They were requested to rate the quality of current training based on the questionnaire items, and also the ranking on the importance of these factors that influence their pre-reg training. This is a sample of our questionnaire. So the outcome measures, as mentioned, was to look at quality rating on the factors and items associated with that influence effectiveness of the training program, and also their perceptions on the importance and impact of these factors in influencing effectiveness of training. The results in terms of response rate, from our preceptors, we had 74% response rate. The trainees, out of 2017, responded for 85% response rate, and therefore an overall response rate of 77%. These are the results. You can see on the left-hand side, arranged according to factors. Okay. The mean score in general was rated higher by the trainees compared to the preceptors. Oops. No. Okay, I'm not sure which is the pointer. No, it's okay. All right, and okay, and the score rated for preceptor-related factors was significantly different between the preceptor and the trainee under the independent t-test conducted. In terms of impact of the factors in affecting our current training, we found that the training program was ranked highest by the trainees and the preceptors. In general, our trainees have also ranked preceptor-related factors as more important than the preceptors. You can see that marked in red. The areas of strength in our current training program, I've listed them according to the mean score of 8 and above. All right, we found that our training program has strengths in terms of its structure and clear training objectives. There were hands-on training opportunities, good variety of cases that can help to develop clinical competence. The training has also helped trainees to apply critical knowledge to real-life practice. Practice to help them to practice safely and effectively, and has met training objectives. All right, what stood out for trainee-related factors was that the trainees in general were receptive to feedback. Other areas of strength related to preceptor, they were willing to teach. They teach, guide trainees in applying knowledge and clinical reasoning. They were inspiring role models. They were committed, supportive, and nurturing of trainees and help them to achieve their goals to gain competency. They have skills and abilities in teaching trainees, and they provide regular and timely feedback for trainees. In terms of environmental related factors, they are living role models in the workplace. All right. For areas of improvement, there were 4. If I could put them together, it's basically 2. All right. One is in the area of sufficient protected time for teaching, and the other for learning, and the other is the current workload pressure that has affected trainees and their preceptors as well. All right. In terms of perceptions on the importance of the factors in influencing effectiveness of training, the trainees ranked preceptor-trainee relationship as number 1, and the preceptors ranked the training program as the most important in terms of influencing how effective training was. Okay, this is represented in a table form. And you can see that trainees ranked the preceptor relationship as most important. In the focus groups, however, which was done earlier on, you can see that actually, it was ranked 5 as the least important, okay? So whilst we are not too sure what it is, our guess is that it's probably a conformance bias because the focus group was done as an activity together. As a conclusion, all right, the factors perceived to be important in influencing how effective pre-registration pharmacist training was. From the trainees' views, it's the preceptor-trainee relationship as rank 1. Number 2, training program. From the preceptor's views, training program is ranked first, followed by trainee-related factors. Okay, and as far as our current training is concerned, both preceptors and trainees have ranked our training program as providing the greatest impact on how effective it is. The score for our training program is 7.34, so it's considered effective. Okay, we have majority of positive responses, 48% of which are rated 8 and above. And the high scores were reported for trainees' receptivity to feedback, the preceptor's clinical competence, good communication skills, willingness to teach, and related to program, that we have good hands-on training opportunities, good variety of cases. Program is also structured and clear training objectives. Two key main concerns were protected time and workload pressure. So what's for the future? Strategies to enhance our current training program and learning experiences of our trainees. Firstly, to do with protected time. This was in discussion with our program director. As per current, actually, our trainees already have half a day per week when they can go off, all right, for their own personal time. So perhaps because they spend quite a lot of time on projects, the program director felt that we could encourage them to use their time, for reflection of their practice. The other was a protected time for preceptors. So going forward, we are going to roster them, for this protected time so that they will be able to be relieved from clinical duties and then be able to meet their trainees for their educational activities. Pertaining to workload pressure, this was an interesting discussion as well, the program director. His point was that if workload is too low, our trainees may not be able to cope with real-life situations. And so, the plan then is to scale up, right, the workload for our fresh graduates, but they will be adjusted up to what is our hospital norm. All right. Preceptors will then also be assigned 1 trainee each time, so 1-to-1 and 1 per year. All right. We'll leverage on IT and automation for pharmacy processes. Actually, we are already very much into that, so I think we'll continue to tap and leverage on that. And the fourth one, an interesting one, which I think was also mentioned by Dr. Ayman earlier on, is about innovative models of training. All right, and this is about blended methods. And in fact, the pharmacy department has in the past 2 years developed topics in the form of lectures and case studies, and then these were posted in a blackboard for the students or for the trainees, so that they can assess anytime, look into them, all right, and be prepared when they come together, all right, the big group with 1 preceptor for case discussions. My concluding remarks: it is important to know about the nature of learning and professional development that takes place as a student transits to professional practice. This transition period, or what we call novice development, is a process of change within the individual in terms of knowledge, skills, thinking, all right, and as well as a contextually grounded professional socialization process. Okay. Clinical learning experiences can shape the formation of professional identity as well as development of professional competence. And this is crucial for novice practitioners as they move to increasing autonomy, responsibility, and greater accountability for patient care and patient outcomes. These are my co-authors. And with this, thank you.
So, do we have any questions?
Yeah, thank you for your presentations. Just I want to know about that because I'm very neighbor to you. I'm from Malaysia, so which I'm working in International Medical University. Regarding this preceptorships, right? You said last point 1:1. So it's including all the government hospitals or in your hospitals only?
At a national level, the ratio that the preceptor can handle would be 1:2.
1:2.
Yes.
It's very nice because of the populations again, I think so. But it's very difficult, like even in Malaysia, which we are facing, even we are handling 10 students is a 1 preceptor ratios in hospitals sometimes. And
So,
one more
so
thing.
1:1 is like, doing very well for, the preceptor. You don't
Yeah,
have to
it's
handle two
national government policies again, right? Yes, because even our government says, because the students ratios, nowadays the pharmacy booming, in Malaysia, right? That's the problem we are facing really. Really the hectic, from morning to the evening, it's very hectic. And two more things I want to ask. What about the preceptor qualifications in national hospitals? In
Preceptor
Singapore, qualifications.
qualifications.
Yes.
The preceptors are actually all practitioners, definitely, in the setting that they're in. The two requirements that are required of them will be, one, they must have at least 3 years of
3 years.
working experience.
Okay.
And then number two, they would have attended the preceptor workshop.
Training workshops, right?
Yeah.
Okay. 3 years after graduations. Okay. If any institutions in your nation are running the post-graduations courses, okay, it's a post-graduation, PG, Master of Pharmacy, anything. What about their training schedules or training programs?
For what again?
For clinical postings. Post-graduation students, clinical pharmacy, post-graduation students.
Sorry, so what about that?
What about their preceptor qualifications?
Oh,
The same undergraduate preceptor, those who are having a 3 years experience, will train the postgraduate students?
Okay, so if it is what we have mentioned is pre-registration, that means those who are fresh
I
graduates.
accept this.
Yeah.
I readily accept.
Yeah.
But
If you're
what
referring to those who are post-graduation,
Yes.
then when they come on board, what is always important is to understand what training objectives they come with. For example, if someone were to come as a PharmD graduate,
Okay.
or someone who comes for specialty, rotation, or clerkship, then we put them through specific supervisors or preceptors.
Doctors are acting as a preceptors, or only the pharmacists are focusing as a preceptor in
Yeah.
Singapore?
If they are pharmacists, then they come under pharmacists for preceptorship.
Okay.
Yeah, so we also have residencies, example, infectious disease residency, oncology residency, and critical care. So for these, right, they are very specific one-year program. There would be a program director with, the faculty members who will then run this program.
Okay, last question. Before that, pre-registration for clinical posting, students are exposing anything with community pharmacy postings?
Repeat that again?
Before this
Expose?
clinical postings, right? Because this is a 1 year posting, 3 and 6 months, 6 months. This is a clinical. I'm asking, your students are exposing any community posting, community pharmacy posting before their completion of graduations?
Okay, so the original program was that they will be exposed to hospital and retail pharmacy 6 weeks each during their student days. But this program will be changed by 2017. They will be doing this as a 3 months rotation in course during the student days, and then there'll be another 3 months rotation, 2 of that post course.
Okay. Yeah, almost similar. Thank you. Thank you very much.
So, any further questions? Thank you very much.
I'm Shinya Uchida from Shizuoka, Japan. So I'm an associate professor in University of Shizuoka. So I come from Japan, Shizuoka, Japan. So many people, everyone know Tokyo and Kyoto, but a few people know Shizuoka. So Shizuoka is located between Tokyo and Kyoto. We can see Mount Fuji from our university, like this photograph. So I'm also interested in pharmacy education because in Japan also pharmacy education is changing dynamically, and we have 80 students a year in our college. But so I'm today I'd like to talk about another interest. I'd like to talk about simultaneous evaluation of the activity of 5-cytochrome P450 enzymes by a cocktail study in healthy volunteers. So cytochrome P450, CYP, are involved in the metabolism of many drugs that are clinically used worldwide. Approximately 50% of the medicines are metabolized by CYP3A4. However, CYP1A2, 2C9, 2C19, and 2D6 also play important roles in drug metabolism. The activities of CYPs are reported to show large inter-individual variation. In addition, certain medications can induce or inhibit their activities, resulting in drug-drug interaction. Thus, determine and predict potential drug interactions is important for dose optimization. There are multiple approaches for studying drug-drug interaction that focus on CYP activities, including in vitro study, modeling and simulation, in vivo pharmacokinetic study using animals, and clinical study, clinical PK study, pharmacokinetic study in healthy subjects or in patient and special populations. Among these various approaches, evaluation by clinical study has the greatest impact. However, multiple drug administrations are required for determining the activity of each CYP isoform. On the other hand, a cocktail approach can measure the activities of multiple CYPs. In this approach, cocktail of multiple CYP-specific probes are simultaneously administered, and the activities of CYPs are evaluated by measuring pharmacokinetics of each specific probes. Since the Pittsburgh cocktail was first introduced in 1797, several cocktails, including Karolinska cocktail, Cooperstone cocktail, and Inge cocktail, has been developed. However, assay techniques in these cocktail studies do not permit the simultaneous analysis of multiple CYP substrate and their metabolites. Thus, in this study, we first aim to develop and validate a rapid and selective LC-MS-MS method to determine the plasma concentrations of 5 CYP probe drugs, and by a single-step extraction followed by a single LC-MS-MS run. In addition, using our cocktail method, we aim to clarify the chronological changes in rifampicin-induced CYP activity after rifampicin discontinuation. First, we developed a general assay that could simultaneously detect all the CYP probe drugs. This slide shows CYP probe drugs and their metabolites in our cocktail study. Caffeine, losartan, omeprazole, dextromethorphan, and midazolam were used as a substrate for CYP1A2, 2C9, 2C19, 2D6, and 3A4, respectively. As shown in this flowchart, in sample preparation, we used an Austro 96 well plate for the extraction of series of CYP substrate and metabolites from human plasma and urine. In the case of urine, the samples were added to the plate after the treatment with beta-glucuronidase. Following optimization of the chromatographic condition, all the peaks were well separated, and retention times range between 4.4 and 11.7 minutes, and total run time for a single injection was within 13 minutes. The precision and accuracy values suggested that the assay has high accuracy and reliability in plasma and urine samples. No significant matrix interference was observed. To demonstrate the clinical application of the assay, plasma and urine concentrations of 5 CYP probe substrates and their metabolites were determined after simultaneous oral administration of 5 drugs to 4 healthy volunteers. All the substrate and metabolites were determined over 8-hour period, and the plasma concentration of each substrate at each hour after administration were above the lower limit of quantification. So we have developed and validated a rapid and selective LC-MS-MS method. Then we aim to clarify the chronological changes in rifampicin-induced CYP activity after rifampicin discontinuation using our cocktail method. 13 healthy volunteers took rifampicin 450 milligrams once a daily, and the cocktail drug was repeatedly administered before the rifampicin treatment, and 24 hours, 4 and 8 days after the rifampicin discontinuation. This slide shows the time courses of plasma concentrations of caffeine, losartan, omeprazole, dextromethorphan, and midazolam after the administration of cocktail drug. After the administration, the plasma concentration of 5 CYP substrate and their metabolites were determined until 8 hours, and pharmacokinetics parameters of drugs and metabolites were calculated from these plasma concentrations. As shown in this slide, the plasma concentration ratio of omeprazole to 5-hydroxyomeprazole was significantly reduced after rifampicin treatment and returned the baseline level 8 days later. Similarly, the AUC ratio of midazolam to 1-hydroxymidazolam was reduced on day 0 and day 3, and on day 7, the reduction was returned. These results suggest that activities of CYP2C19 and CYP2C3A4 were induced by rifampicin treatment, and the inductions remained at 4 days and returned 8 days later. So in conclusion, we developed and validated a rapid selective LC-MS method to determine the plasma concentrations of 5 CYP probe drugs and metabolites by single-step extraction followed by a single LC-MS run. This assay had high accuracy and reliability for plasma and urine samples. And the induction of CYP2C19 and CYP3A4 activities by rifampicin remained at 4 days after the rifampicin discontinuation and returned to baseline levels 8 days later. So our cocktail approach enables in vivo assessment of the activity of various drug metabolizing enzymes and the detection of potential drug interactions in a single assay. These are the people with whom we have collaborated. Especially, a clinical study was done in collaboration with Professor Watanabe and Dr. Inui at Hamamatsu University School of Medicine. And Shimako Tanaka mainly performed the development of LC-MS method. Thank you for your attention.
So, are there any questions? I unfortunately got your presentation halfway, but I'm interested in the method that you use to determine those metabolites, because I work with coffee and I look into caffeine and I look to genotyping and the CYP1A2. So, and I'm exactly right now going to this gold standard method to measure metabolites. So, do you have your method is already published to do that you validated and all that? So, the LC-MS method?
That report was published already, yes.
And is that one of those that you put on the bottom of the screen when you were showing the data? Was published in that magazine that was showing the bottom of the screen?
Yeah.
International something. Can I get that from you, the reference? I'm interested in the reference. Yeah.
Okay, so assay method was published in this.
Okay, 2014, all right.
Mm-hmm.
Thank you so much.
Thank you.
Thank you. Are there other questions? No.
Thank you. Thank you.
Thank you very much.
I'm sorry that there's a lot of the speakers which wasn't able to come here. This was actually my keynote speaking, my keynote presentation, and I want to keep it now. And this is related to treatment of cancer. And the reason why this is important is that this new substance, which is on the level, stage 2 level, is important, is because it is a special substance which could be used in usual court. The contents of this substance is a combination between cytostatica, cisplatin, but it is mainly working directly on the immune system, so it's immune stimulating component combined with low value of cisplatin in a reaction. That means that it works in the mechanism action in this card is very different from the one we have seen before, and that's why I'm together with a lot of others in developing this worldwide. The main part of this is actually performed in Asia. We have used the dose response studies performed in Taiwan, in Thailand, in Mahidol University Hospital, and in Indonesia. And it's also performed a dose study in Europe, but there is a lot of Asian patients. The reason why I'm so interested in this is because this is a substance which can be administered in usual healthcare system. It is not related. It's classified as 2. So it can be administered outside the hospital, and in a lot of these countries, not these I mentioned, but in Cambodia, Laos, and so on, they don't have so much available treatment for cancer, and they are to rely on Thailand, most of these patients. This can be administered in the forest, so to say, and by nurses or pharmacists, if they have pharmacists. It's a combination, and it's given intramuscularly. And what I'm going to show to you now is the results which we have obtained in in the latest study. It's quite complex, this design, but it's a multi-center study in Europe and Asia, because it's some Asian patient also included. If you look upon this, the patients are stratified with regards to age first, less than 45 years of age, 45 to 60, and past 60. Within these groups, the patients are randomized either to treatment with this active substance or equal-looking placebo. So it's a placebo, double-blind placebo-controlled study. But due to the fact that the ethical part, the patient which is allocated to placebo would be crossed to BPC1 after the first cycle of 32 days. So the BPC1 patients which respond will then can continue the treatment, or they withdraw from the study. The patient which have been allocated to placebo will be crossed to active treatment of BPC1 or withdraw from the study, and then they can continue with additional treatment. The reason why we are using such kind of design, this is a semi-crossover design, half semi-crossover design, is related to the ethical aspect of performing patient with this kind of disease into placebo treatment. So it's actually a prolonged start of the treatment instead of saying that it's placebo. If you looked in, this is the patient which is included here is stage 4 breast cancer patient, which means that I have distant metastasis, and all of them is actually undergoing surgery, so the breast or the tumor is removed, but this is a treatment of the metastasis. The point is that all these patients have passed through at least 4 different kind of cytostatica course before they went into the study. So they are very difficult patients to treat, actually. What we obtain here is that we have 15 patients, so it's not a big study, it's a huge, small study, 15 patients in each group, 15 patients which get BPC1, and 15 was randomized to placebo. And you see that the age here is the age and the duration of the disease is quite large. It's more than 6 years, which means that they have been treating for this cancer for several years. And there is no difficulties with the systole, with the blood pressure or vital signs. And as you can see here, all have undergone surgery. They have got hormone therapy, most of them. They have also radiotherapy, and some have other therapy in addition to that. So it's a different group of patients to treat. If you look into the results here, we can see that this is a tumor, the metastasis, the lesions, target lesions in accordance with the RECIST criteria. And if you see at the BPC1, there is no change in the tumor. It's only an increase of 2.4%. If you look into placebo group, you see an increase of 14.3%, which is significantly. And there is no significant increase in the BPC1 group, but there is a significant increase in the placebo group with regards to the size of the tumor. So this is an obvious indication that this new substance working directly on the immune system, but also at the killing part by using cytostatica. And the point here is that the cisplatin is actually accumulated in the tumor itself. And the reason why it can be that is that this BPX1, its main substance, is then influencing the cancer cells. So the cisplatin can penetrate into the tumor itself and accumulated into the tumor until the concentration is so high that they kill the cell. If you look upon the classification here, this is the international classification system, but you see that there is a significant difference. We have one partial responders and 14 steady state patients. Here we have 4 which have a progressive disease and 11 which have stable disease. So the difference is not that big, but it's still significant in accordance with the criteria which is used. If you look upon the quality of life, I will focus on this one here. Quality of life, which is measured on this C30 schedule, and this related the quality of life, which is related to breast cancer. And if you see on the breast cancer, pain related to and discomfort related to the cancer, the last week we can see that this pain and discomfort is reducing in the BPC1 group, and it is actually constant in the other, but there is a significant difference between the two groups with regards to quality of life. So it's indicated that this is directly working on the immune system, and the patient feel better, and the quality of their life will increase a bit in accordance with the treatment. If you look at the bone, this is a very interesting thing, because what I've seen before is that the toxicity score, the first time I obtained that was that the toxicity was actually negatively correlated with the duration or the amount of substance, and it's positively correlated with the reduction in tumor. So it seems that the more drug you give, the less toxicity you obtain, and that's horrible. You should not obtain that, because you're giving a toxic drug to the patient, and the toxicity going down. So why? Well, the reason behind that is that when we are starting the treatment with the patient, the body itself is very intoxicated by the tumor itself. So it's on a certain level. So when you give this intramuscularly drug for a certain period of time, the toxicity go down, which is not a proof that this is untoxic. It is toxic, but the toxicity which is given by the drugs is less than the toxicity caused by the cancer. So the toxicity of the cancer go down, and that go more down than the increase caused by the drug itself. A very strange situation. So, and this have been seen before in single patients where we are giving cisplatin, for instance, and we see that the toxicity and the quality of life of the patient will increase, and the toxicity go down, and the patient is responding. But in the main case, what's happened when you gave cytostatica chemotherapy is that the toxicity increasing and the patient feeling bad and awful, and the adverse events increase and so on. That's a general situation. But the patient which responds to the treatment seems to have another pattern than the general situation in cancer treatment. So, and if you see here, you can see that in the BBC1 group, there is an increase in average, increase in the toxicity by 13.6%, but in the placebo group, it's increased with 27.7%. So the increase in the placebo group is nearly double what you get, more than double of the one which you get in placebo, which give the results in the same direction as we have seen before. If you look into the pattern of adverse events, by treating patients with cancer, and especially stage 4 cancer, you know that the list of adverse events is very long, and they are very, the severity is high. It's moderate or severe, and even worse than that. If you look upon the list here, you can see that this is nausea, it's headache, flushing, fatigue, and so on, but all of them is mild. There is some of them which is moderate, and that is related to the laboratory findings, which I will present later this Congress. These are very low, and they are going away after a while. And there is one adverse event which is very strange, because some of the patients is reporting that they feel that they have got the flu. They have the feeling that they got the flu with the symptoms of the muscles and so on, and the day after it's gone. And then when they have continued the treatment, after, let's say, 1 week, they got the same symptom again, and they disappear. So the question, I was finding that in several patients, and the question was why. And the reason is that the level of interferon is actually increasing 8 times by this stimulated 8 times. So it is actually seems to be the interferon level which is increasing, and that was causing the adverse events of this kind. So in conclusion, I can say that this is actually a very interesting drug, and it's an interesting drug because it can be given in outside the hospital, in healthcare system, in a population where we don't have the needed specialities and the needed way of following up these kind of patients, so it can be given to all the patients in a very proper manner. And it's shown that it has a positive and significant effect on the tumor or the metastasis during treatment, increasing the quality of life, and it reducing the degree of toxicity, which is very interesting to show. And it is very few side effects, adverse events, and mainly they are mild, and they are going away again during a period of time. So it's not an adverse events which will stay there for a long period of time. I've found this substance so interesting that I have continued working with that, especially because it's related to patients with this dangerous disease in the poorest part of the world. And I see that it is very important for a researcher also to do something for these patients itself. Thank you.
So, are there any questions?
Okay, sorry, I didn't see you. Thank you for the wonderful presentation. And I wonder, I think I missed when you said, what is the posology? I mean, are you administering intramuscularly? That's what you said,
It's
right?
intramuscularly.
And it's like 0.5 mLs?
No, the dosage is 0.07 milliliter per kilo body weight. So it's administered per kilo body weight.
Okay.
Yeah.
And that's why you can do outside the facility,
Yeah.
right? Uh-huh.
Yeah, also because, you know, when you give chemotherapy, there is special rules of that.
Right.
You have to be covered and all this kind of thing,
Right.
because it's dangerous. But this is not dangerous. You can take it in your hand, it's no problem.
Right.
And that is quite amusing, because such kind of drugs is very dangerous, and you cannot use that outside a specialized part of the hospital.
Okay. And during those 32 days that you follow up the treatment, how many times the injection was given?
They got one injection every day, daily injection.
Daily?
Yeah.
Okay.
And you know, the reason it's me. I've used the results obtained from the preclinical results, and see that the preclinical results indicate that you shouldn't treat the patient more than 32 days,
Okay.
due to the fact that you can get adverse events, serious adverse events. But we found that this didn't happen, and today there is. Several patients which is actually going on the treatment because they respond. We have especially one patient in Russia. She has been treated now with this drug for 26 times 32. It's nearly 3 years. And what's happened with her is that she was said to be dead, actually. And the tumor, the metastasis, was going down, so it's a partial responder. It's only 10% of the tumor left, and she is have a very good quality of life, so she wants to go back, but we still the doctors are following her up. We have one patient in Thailand, which has now been treated for 12 times, nearly 2 years, which is also responding in the same way. So it seems to be very well tolerated by the patient. The patient feel that they are better themselves, and it works directly to the tumor.
So do you have any data in terms of increase of life expectancy?
No, I have some data which we have used for analysis to estimate the time to progression.
Okay.
But you know, this is not a big pharmaceutical company running that. It's a university developed system, so we don't have so much money. So the number of patients are quite low, and we haven't been able to follow them for so long time. And the problem, you know, with time to death in this is that if you stop the treatment, the doctor will continue with a new treatment, and you don't know if you're prolonging the life of the patient, if that is caused by the treatment you have given or the treatment the patient have given after.
Okay.
So no, we don't have the data.
All right. Okay. Thank you.
Thank you, Rosemary.
So, are there any other questions to this? Okay, thank you very much to Professor Stig Larsen.
So I think I have a chance to talk for almost 1 hour, but I'm trying to get it sooner because we have lunch. So I'm quite excited to go for the lunch right away. So I will try to wrap things as soon as I can. But I think this is a very important topic that I want just to give you an example of research that is based on clinical practice. This is what we call it translational research. And this is a new approach that I concluded in my lab and in my research activity to try to come up with what we call it, the personalized medicine based on different factors, different doses, based on different disease states, and maybe some genetic factors also. In this case, I'm an antibiotics person, so I'm an infectious disease person. So when we talk about pharmacogenomics, I'm not dealing with the CYP and the metabolism. I'm dealing with the genes in the bacteria itself, because this is a target. So I'm more interested to identify the genes that are responsible for resistance of these organisms and try to find a way to overcome this resistance by modulating and changing the dosing. So my ultimate goal is to make sure that I achieve the maximum dose based on what I have here and try to make sure that I achieve this dose by simply maximizing the therapy. Let me see, this is the right one. I usually do that when I have some research presentation. I start by a case study just to give you the stage, set the stage for that, and then I give you the rationale for that research. Then I will give you the real beef in that research, which is basically my specialty, which is PK/PD, pharmacokinetic pharmacodynamics, and in my case, it's more of antibiotics PK/PD. And then I give you example of my approach or our approach that we used to have it for the last 10 or 15 years now, together with my colleagues who started this model. And I can't forget, Dr. George Dursano, George Zanel, all of these guys are great, who helped me to learn, and all of us tried to do something in the field of PK/PD, achieving the goal of maximizing the dose of antibiotics. This is part of the fight or the war against bacterial resistance. Let me start by a case here, and this is classic. I do that all the time. FT, whatever the name is a 25 years old male with 2 months history of cough and increased sputum production. He presents to the outpatient clinic with fever, new chest pain. When he coughs, he has shortness of breath, blood-tinged sputum. He has history of bronchial asthma since childhood. He has sickle cell anemia that required splenectomy. When I say splenectomy, this means what? That we remove the source of production of the immune system. So this patient is almost immune compromised. So this is one factor here. The medication history is basically therapy for the, let me see if I have pointer here. Nope. Kills it. Yeah, it works. So this patient have history of medication for asthma, salbutamol inhaler to pass QID, and he has also beclomethasone inhaler. These are classic for asthma therapy, and he receives oxycodone and aspirin to tablets Q6 hours PRN. This is for the serious, severe pain that sickled patients are having or going through. But also what's important here that this patient had history of ciprofloxacin, 500 milligram Q8 hours for 10 days, 1 month ago. We are not quite sure what was the reason for that, but somewhere, somehow, within last months, he went to a clinic, and I think the physician, he had some whatever infection, and it was maybe needed to have ciprofloxacin. And my wild guess that this was simple, pulmonary or lung infection, that it might have been even flu or maybe simple laryngitis, whatever. But the reason here is simply to mention that this was a history, which is very important here. That's why I highlighted that this was a ciprofloxacin, a fluoroquinolone received 1 month before for some reason. This patient was diagnosed with community-acquired pneumonia, CAP, that was a PSI score, severity score of 3. This means that this is mild to severe pneumonia. For sure, he was admitted to the internal medicine ward, and he received the classic therapy at that time, levofloxacin 500 milligram Q 24 hours. This is classic case, patient who was admitted with pneumonia. We gave him levofloxacin, and this is the used gold standard therapy right now. Then a culture was made, and it was found to be Streptococcus pneumoniae. He has MIC initially was 2 microgram per ml that had PAR-C gene. Then within therapy, within few days in therapy, this MIC jumped from 2 to 16. So now the resistance began to be more evident here, and now we have 2 genes responsible for the resistance. This patient received medication, had Streptococcus pneumoniae. However, this should be a successful story, but there is no improvement. Patient got meningitis, developed meningitis on day 4 and died. So this is a story of a patient who died. And this is a really a dire outcome that we don't expect, but there was something wrong here. Because this patient supposedly should not die, because he is young, 25 years old. Yes, he is immunocompromised a little bit, but he is healthy. And he has come here with a simple infection, community-acquired pneumonia that can easily be treated by levofloxacin. And this therapy will stay for last for maybe 3, 4, 5 days and send home to continue for maybe 2 weeks. And that's it. And he should be recovered. But this is a patient who died. So there is something wrong here. And as clinical pharmacists, as clinicians, as scientists, we should not stop here and say, okay, he died. Who cares? Let's just see what's going to happen. Our duty is just to go back and say, what happened here? What was the story here? And begin to learn from that and identify the problem. What was missing here? You know, this guy received medication that's adequate for therapy for a simple treatable disease, and he's a young, healthy, patient. So what will happen should be a successful outcome, but he died. So our duty and our values dictate that we have to go back and stay and pause and say, there is something wrong. What happened? We have to learn from that. So, and this is also as a scientist, as a translational scientist, I have to identify the problems in the clinic here and learn from this problem and begin to identify the reason, find a solution, and then go back and try to help saving lives after that. So to do that, we had to go back to the basics. We have to go back to the textbook and say, what I learned from this story here. I'll go back to the basics. I'll say, let's just go back to that day 1 when someone in the lab discovered a molecule which is antibiotic and claimed that this is going to be effective in treating or killing bacteria. And we gave it to the patient, and the patient actually recovered. But does this happen all the time? No. Most of the time, we give the ideal molecule that was identified to be a good antibiotic, and you give it to the lab, and the lab will show at the lab level that this is really effective antibiotic. It kills all the bacteria. I can see it under microscope. I give to the patient. It doesn't work. It happens every day. We see that every day. So this is a story that we have to stop and say, hey, what's happening here? Let's learn from our experience and background. Let's think about it. What are the causes of this antibiotic failure? Tons of causes. So I'm not going to go over all of these causes, including maybe compliance, all of the things, but I will focus here as a clinical pharmacologist or clinical pharmacist in what's really important for me that I can learn from that. I'll focus on the pharmacological failure and also focus on the microorganism itself. These are the 2 components that as a pharmacist I'm really concerned. What will be my enemy, the bug? Is it a dangerous enemy? Is it resistant? And also what will be the effect of my therapy, the pharmacological effect? So these are the 2 components that I'm really interested to look at. So I look at the pharmacological effect and see maybe the drug given in an insufficient dose, or there was no real good diagnosis, or there might be something wrong in the pharmacodynamic parameters. There might be something that led to a suboptimal therapy. At the same time, there might be something wrong with the pathogen itself. It might be resistance that developed during treatment. I have to understand that. Or maybe the antimicrobial is giving there, but it doesn't have good coverage for the bacteria, and maybe there might be some bacterial persistence. So these are the factors that I have to highlight and look under the microscope and try to understand what's happening here. So I go back to the microbiological evaluation. What happened is that there is something wrong here. What happened is that I receive information based on a static model. Let's put it this way. So in the lab, what they do, they have all of these antimicrobials, plates, the plate with the bacteria there, and then we have all of these discs for different antibiotics, and then see what's the sensitivity, and then try to identify the MIC and the breakpoints. This is typical microbiology lab, maybe microdilution, where I can see the turbidity, and then I try to define the MIC. This is what the lab does all the time, till now. But tell me, do you think the human body functions this way? Are we static thing? We are dynamic. Everything is circulating. We have dynamic process. There is blood circulating all the time. You give the drug, it goes into the absorption, and then you reach the peak, and then the elimination. It's a process that goes to a dynamic process. And also, there are different factors. If I get a drug, antibiotic, my friend who is 25 years old get the same antibiotic, is it going to be the same effect? No, because I have different physiology. I have different kinetics. So this is different stories. It's not a plate with an antibiotic with a bug and try to see what will be the outcome. So what I'm arguing against right now is that what we are doing here is absolutely wrong. It doesn't represent the real clinical setting. So our job as clinicians or scientists in this case is try to model what's happening in the real setting. So we can come up with outcome that's really representing what's happening in the real human body and in the clinic. And another point to argue against that is simply when somebody say that this is basically the effect, the drug given over time, the concentration is here, but then you talk about MIC from the lab. Where is this MIC? Is it here at the peak? Is it here at the trough? Is it here at the middle? Who knows? So that's why this is really a solid argument against what we are talking about initially as a static model. This is garbage. We should not rely on that. We have to rely on more of dynamic modeling of what's happening. And this is what I'm going to build the case right now. And that's what we tried to do that maybe 15 years ago. I joined this group maybe after 5 years of their work, and I began to work with them. And the idea here is simply that we have to combine the pharmacokinetics and the pharmacodynamics of the antibiotic both together into consideration. We have to get them, I call it marriage, where we combine all of the effect of the drug on the human from the kinetics point of view as absorption, distribution, metabolism, excretion, again, together with the pharmacodynamics, which is basically the effect of the antibiotic on the bacteria. Like, talk about potency, killing, persistent effect, and immune factor system that will have a component in this type of effect. What we do here is that we have to include and consider and combine, I call it getting a marriage certificate between both two factors here to aim toward bacterial eradication. So my theory here, or what we propose here, is that we have to put into consideration the kinetics and the dynamics, both of them, in making our decisions for dosing and optimizing the therapy for these patients in antibiotics. I'm talking about antibiotics here. That's why when we have this classic, concentration versus time, curve, the bell-shaped curve, which is very famous, when you have the drug given and then you go through the absorption, and then you have the distribution, and then you have the Cmax or the maximum concentration, and then we go through the elimination phase where you have the drug concentration decline in the body, you give the second dose, the third dose, and so on, and this is where we have the kinetic parameters, or the kinetic parameters, where you have the Cmax and Cmin, you have area under the curve, we have a plot for the sigmoid, a structure there that you can have the elimination rate constant, and you go ahead and calculate all the parameters for the VD, volume distribution, all of these things. This is kinetics. But as I said, it's not only kinetics. We added dynamics into it. So now we have a new way of looking at the drug effect here. When we have the kinetics, but then add to it the dynamics, which is typically in the case of antibiotics, the MIC, the minimum inhibitory concentration. So you have to consider the MIC for this bacteria. It might be 1, it might be 2, it might be 10, it might be 0.01, whatever. But I have to consider the MIC in my calculations. I have to consider it in my predictions of outcome. So here we put the MIC. Now we came up with some new principles that dictate or which shows us exactly what are the most important parameters that will govern the antimicrobial effect. So we came up with new things here. We call it Cmax divided by MIC. This is one factor. AUC, which is area under the curve, divided by the MIC, another factor. Then we come here with the time above the MIC. So this is the time, let's say from hour 1 to hour 12 or hour 13, the time the drug spends above the MIC. 13 minus 1 is 12. So the drug spends, let's say, 12 hours, 4 hours, 20 hours, whatever. But these are factors that we have to consider to make sure that these are going to govern the outcome of our therapy. So let me give you an example here. And by the way, this is a work of great guys, Alan Forrest, talk about Mike Rybeck. All of these guys spend years and years in the lab and working. I did some part of this work also in my lab when I was in Canada, that we did things with Streptococcus pneumoniae, and this was against the macrolide. But in this case, I did some work with the fluoroquinolones also. Let me just focus as an example. But this example can be applied in everything. So when you say fluoroquinolone, you can put here, let's say, azithromycin. You can say penicillin. You can say cephalosporin. This is one factor. So sky is the limit. Research is unlimited here. Versus Streptococcus pneumoniae. It can be MRSA. It can be Acinetobacter. It can be Pseudomonas aeruginosa. You name it. So the variables, you can play with it. You can change it. But each one is a story by itself. So let's talk about a story here of, and especially this is related to our patient. So I chose a patient who has Streptococcus pneumoniae, remember? And this is a community-acquired pneumonia infection. And this patient was given fluoroquinolone. So I'm trying to solve the issue here. So luckily, those guys like Zanel, Andy and Greg Peterson and Alan Forrest and Thomas Lodis, all of these guys did lots of work. And still, you talk about Kuti, you talk about David Nicolo, all of these guys are still working. We are still working in our labs to try to come up with different scenarios and make sure that we are doing a good job. So at the end of the day, at least especially, I know that it was mainly Alan Forrest, we call him the bear, who came up with some real nice, and I think Shintag at the same time, came up with some real numbers. And this is cool. When you have numbers, you say, yes, if achieve this target, you can kill the bacteria. Don't achieve it, no way. So came up with numbers here after lots of experiments. I'm going to show sometime maybe in the next lecture how they did that. They had lots of animal models. We developed lots of in vitro models to come up with real numbers that will say, hey, when you have a patient who has nosocomial pneumonia, and you find that the fluoroquinolone area under the curve divided by MIC, whatever the number is, if you divide that and you get a number of 125 or above, you are successful. You are going to achieve bacterial eradication. If you achieve AUC over the MIC of, let's say, 90, it's not enough. What this means? This means that the AUC area under the curve, the amount of the drug that was given is not enough. So what's the message here? Increase the dose. This tells me that the dose is not good enough, increase the dose. Or the other side, if the MIC is 1, that's fine. But if the MIC is 2 or 3 or 4 or 5, this means that the outcome will be lower. So in this case, the more resistant the bug, the more difficult to treat. So in this case, in the case of fluoroquinolone, you have to increase the dose to achieve, in case of nosocomial pneumonia, AUC over the MIC of 125. When I say AUC here, I have to talk about the free drug, because the free drug is the active drug. So I have to include also factor for the protein binding here. So I multiply by 0.07, whatever the factor here. This is in case of nosocomial pneumonia. In case of our patient community-acquired pneumonia, they found that area under the curve of the free drug, when you divide by the MIC, your target, we call it target attainment, is 30 or above. So this is your target here. As a clinical pharmacologist or clinical pharmacist, you have now tools to calculate and predict the outcome. Let me give this drug. Let me calculate the AUC for the free drug. Let me look at the lab and tell me what the MIC is. Let me calculate the AUC over the MIC. Is it 30 or above? Yes. Bingo. I know that this patient will be treated. But if AUC over the MIC is less than 30, this patient is going to die. Period. So this is what we call it prediction based on numbers and principles, not just guessing. And this is what's happening in the clinical setting. Everything, especially in antibiotics, is a guessing. Let me give him this drug. Oh, it didn't work. All right, just increase the dose. How much? I don't know. Let's increase it a little bit up. Or reduce it a little bit. Or maybe give another drug. Oh, let's just stop it and give another drug. Based on what, my friend? Nothing. It's just, oh, this is my experience. No, this is garbage. I need facts. I need numbers. That's why when we have our rotation, we calculate that and say, hey, my numbers, my calculations show me that your dose is not right. You need to increase the dose or change the medication. How did you come with that? My friend, these are the numbers, these are the facts, this is my calculation, and now I tell you that this is going to work if you adjust the dose based on what I tell you. So let's just go back to the fluoroquinolone and to our patient. Now we are done with the pharmacological part here. Let's go to the other side of the story. We said that the two components here are the kinetics and the dynamics. Now we are done with the kinetics. What about the dynamics? Because the dynamics is basically MIC, which is basically dependent on the bacterial resistance. If the MIC is 1 or higher, this is more resistant. Let me look at that and understand what's happening here. And this is the other side of the story. My enemy is a smart guy and very clever and actually changing the pattern of resistance every moment. And dependent on very factors can sneak in and try to find a way to beat my therapy here. So I have to be careful. So to do that, I have to understand what this bug is doing here, what the mechanism of resistance. To get the mechanism of resistance, I have to learn first what the mechanism of action of that drug that the bug began to develop resistance against. So let me just understand that. Let me go back to my literature and say, okay, how this fluoroquinolone works. They work mainly on the topoisomerases. It binds to those topoisomerases, and you have DNA gyrase, which is topoisomerase 3 or topoisomerase 4. And the fluoroquinolones work by just modifying these topoisomerases that are very important to unwind the DNA. And by the way, this topoisomerase 4 works by action of the gene or stimulation of the gene PAR-C or PAR-E. In our case here, it was found that PAR-C is the more active. This is the one that I have to worry about. For the DNA, topoisomerase 3, which is the DNA gyrase, they have gyre and gyre-B. Gyre-A is the one that's more active. So this is the guy that I have to worry about. So here, the genes that are on the DNA or for these topoisomerases are responsible for encoding for the topoisomerase and production of topoisomerase are the PAR-C and the gyre-A. So I'm a bug. I understand that the topoisomerases are the ones that are important for, for my life. So what they do, the antibiotic, what does is simply comes here and work on this PAR-C and gyre-A and the topoisomerases and try to modify it and doesn't get it. So now I stop the replication of the DNA. Now I can kill the bacteria. As a bug, what I do? Let me fool these guys. The fluoroquinolone comes here and try to destroy my topoisomerases by working on the PAR-C and the gyre-A. So what I will do? I will change the gene structure here. So I'll try to express the PAR-C and work on the gyre-A and try to make it more prominent. So the fluoroquinolone comes here, you don't find it. They find a modified
genes. And the antibiotic comes here, find nothing to treat or nothing to kill or modify. That's why the antibiotic doesn't work. You just give this antibiotic as water. Doesn't work. The target is not there. They fool the target. And this is a very smart way the bacteria do all the time. I'm just giving you an example of Streptococcus pneumoniae when you treat with the fluoroquinolone. There is lots of other mechanisms like the MRSA, like Pseudomonas. They have tons of things. But our job is to identify this mechanism of resistance and study it. And then learn from this mechanism of resistance and begin to try to tailor our therapy to be smarter, hopefully, than the bacteria itself. So this is exactly what happened. The resistance here, because there is now the bacteria begin to modify the binding sites, the gyre and pyr-C. So the antibiotic comes here, find nothing. It's different, it's modified, so no effect. The other mechanism is simply by the bacteria. The antibiotic comes to the cell of the bacteria here, but then there is a pump, efflux pump, PMRA gene based, that will simply sweep or clean the antibiotic out, push it out. And this is other mechanism, but by the way, this mechanism is not that effective. So mainly target modification is the key for resistance in Streptococcus pneumoniae versus fluoroquinolones. So this is resistance that's happening. Another characteristic of this resistance is very interesting. We have to study it. It is rapid during therapy. So you can give the antibiotic, the fluoroquinolone, to a bacteria or a Streptococcus pneumoniae with MIC of 1, which is not resistance per se. But during therapy, it can switch and change the genes and make it modified, so the bacteria will have different genes to work on for the fluoroquinolones. Now the fluoroquinolone come and find no target. The bacteria begin to have more resistance. So the MIC can jump from 1 to 4 to 16 during therapy, which is far more dangerous than what we expect. You can start with a real, non-resistant bacteria, but then during therapy, it changed to be resistant or development of resistance during therapy itself. The other problem here is that it's spontaneous. There is no extra genes to get outside from other bacteria. It is there. The genes are there, just modifying the genes. It's a spontaneous process. And also what's more important is that it is stepwise accumulation, i.e., it starts first by modifying one gene, the PAR-C. So the MIC jumps from 1 to 2 or 3 or 4. This is called low-level resistance. Still can be treated by fluoroquinolones. But then, during therapy, if you still keep giving the fluoroquinolone, this resistance jumps to the second step or next step, which is PAR-C gyre. You have both genes are modified. Now MIC jumps from 1, 2 to 16, 32 or more than that. This is high level of resistance. No way fluoroquinolone will work here. So this is too much for the fluoroquinolone to work on, unless we change the dose here. But this is what happens. It's a two-step, PAR-C and then gyre. So now we understand what happens for the drug in the body, the kinetics. Now we understand what happens for the bacteria, for the resistance. Now let's just get some lessons and see what we can do to identify that, and then what we can do to overcome that. So we thought about it, and we said, okay, our first step is to identify bacteria that has first step mutations. Because you see, ciprofloxacin or fluoroquinolones are in the air, in the tap water. You go to a physician just for simple laryngitis or simple flu or simple symptoms, upper respiratory tract infection, and just give you ciprofloxacin or give you a fluoroquinolone. And this is normal practice or abuse of therapy or prescription pattern all over the world. This is one thing. Here's the point. When you give a patient ciprofloxacin for something else, what happens is that the normal flora, the normal Streptococcus pneumoniae in the throat is affected. So it finds that there is ciprofloxacin there. Oh, wow, let me change into a resistant organism. So they change into a first step PAR-C level of resistance. And our hypothesis was simply that it is there, but we don't know it. It is there, but we don't have any idea what's happening here. So our job is to detect the first step mutation and see how things are working there. The second step is just to try to find a way to kill this first step mutation before it is too late. Because if it goes to the second step, MIC is very high, it will be very difficult. So our way of preventing is to identify the first step mutants and try to wipe it and get rid of it. So this was our approach. How did we do that? At that time, I was working in Canada with George Zanel at that time, and we noted that levofloxacin, reported to be successful in the lab, but then when you give to the patient, there is still no good outcome. So the question here is that those susceptible organisms, are they really susceptible? We receive from the lab, SIR. It's susceptible, it's S. But is it really susceptible? We question that, because the way they test it is not really adequate. This is what we need to go at the gene level and identify, because it might look susceptible, but it have a PAR C first step mutation. This was our hypothesis. So let's do that. So how we did that? We still run that till now. We run North American surveillance study. So we collect isolates from all over North America, and now we are expanding that to different parts of the world right now. And there's lots of studies like that, where we collect all streptococcus from the hospitals that are isolated from patients with community-acquired pneumonia, for example. And we get all of this pool of bacteria, and then identify the MIC, and then see what the MIC there. But what's more, we just do genotyping for those resistant ones. So if we find one which is higher than what we believe is normal MIC, we begin to identify the gene there, identify the PAR C and the gyre and all of these things. So we do some genetic identification. Beyond the MIC, we just identify the PAR C and gyre mutants in our isolates. And here's what happened. Believe it or not, we discovered that out of thousands of isolates that are tagged as susceptible to fluoroquinolones, we discovered that at least 70% of those susceptible isolates, they carry PAR C. And then the rest of them, maybe 3% gyre, and then maybe PAR C gyre. But what's important here is that those isolates around us that has, simple susceptibility, which is simply susceptible, are actually having PAR C. This is very important and very dangerous. Somebody tell me why. Because this patient who has this PAR C isolate, if he or she happens to receive a fluoroquinolone, what will happen? It will jump to the second stage or second step of resistance is going to be PAR C gyre. PAR C gyre is a high-level resistance. MIC will be 16, 20, 33, whatever. This is really a very difficult situation. So this is very important. That's why we called it the tip of iceberg. There is something there, but we are not quite sure that there is really a big thing there. There is something that we have to worry. This is really a major problem. This is major public health issue here. So what to do? We detected the first step mutants and we identified the iceberg. Then the second step is to try to identify the doses that can kill bacteria with first step mutations. And maybe hopefully the second step also, if we can approach that. And this is what I call it clinical simulation and pharmacogenomic approach. How we do that? We cannot test it or test drive it on patients. We cannot bring patients and infect them with PAR C gyre or organism and then give them medication and try to see who's going to die, who's going to survive. This is not going to happen. And also we cannot, test it, in different even animals. So we thought that we do what we call PK/PD, clinical simulation. Let's just do simulation and mathematical modeling to identify the scenarios that will be successful to be able to eradicate this bacteria. So this is what we did. We built pharmacokinetic population model. And this can be built either from running different clinical studies, simple on healthy volunteers to identify the kinetics of fluoroquinolone in patients or in humans or in healthy volunteers. The key thing is just we identify the kinetic parameters. But we build a kinetic model, population model, that will represent a big population. So we don't say that we have 5 healthy volunteers, we give them drug A, and then we measure the drug, and then we found that the PK is whatever, the Vmax or the Tmax or VD or the AUC is so and so. This represents 5 patients. It doesn't represent the whole population. So what we do is in this case, what we did, we collected different data from different studies all over the world. Those guys who published, kinetics of fluoroquinolone, let's say levofloxacin, in patients, whatever. Let's just get all the data from them and put it into a model. We call it pharmacometrics or pharmacokinetic population model that basically represent the overall population. You talk about the whole population, and you can make simulation to make it represent thousands of patients. This is more reliable, more representative. So I can say that the VD, volume distribution of fluoroquinolone or levofloxacin, and those patients who are 20 to 50 years old is that number. And I'm quite sure that my number is accurate because it represents huge bell-shaped data. So it is really all over the place. The same thing I do it for the MIC. We got all the MIC from those pathogens, identified the PAR-C, and then we calculated all of them. You talk about at least hundreds of MICs. Is it enough? No. What we did, we have the kinetics distribution of data, and then we have the dynamics, which is the MIC distribution of data, and then we got them married. That's what I said, PK and PD marriage. We added both of them to build that model, and then we ran what we call it Monte Carlo simulation. It's a Markov model. It's simply iteration, so it will combine both data here and then begin to build what we call it what-if scenario, based on real kinetics data, based on real dynamics data. And that's exactly our model. That's what we call it clinical simulation. So our hypothesis is simply that those drugs or fluoroquinolones that have higher AUC, you remember that the factor there is AUC above the MIC, so the higher the area under the curve, the higher drug exposure, the better effect to kill these bacteria. And that's what we did. So we modeled AUC distribution of Cepro, Levo, Gatti, Garinoxacin, Moxifloxacin, Gemifloxacin. At that time, these are the most important ones. Even some of them were research-wise, but we want to have the whole spectrum there. And then we modeled different treatment scenarios, modeled different dosage. So Cepro, if it's given to 50 once daily, 500 once daily, BD twice daily, 500 BD, 750 BD. Levo, what are the doses? Even I put here 1,000. It's not really clinically relevant, but I thought that let me just see what if we give 1,000. It's not clinically relevant, but let me just, because this is a hypothesis. So I ran from 500, 750 to 1,000. Gatti, I just modeled 400 for all of them, which is the clinical dosage for that. So we modeled what if scenario. If we give the dose of levofloxacin 500 milligram, what will be the area under the curve from my PK model? And if I give a dose of Moxi 400 milligram, what will be the AUC for my model here? At the same time, I have all the Streptococcus pneumoniae genetics distribution there. I combined both of them into a Markov model. So I got all of these AUC distribution versus MIC distribution into a Monte Carlo Markov modeling there, and then the outcome will tell me what's going to happen. If I find that AUC over the MIC, as I said for the fluoroquinolone, is more than 30 or above, this means that my therapy in that dose is going to be effective. If it's less, it's not going to be effective. So now I have numbers that I can work with. So what I did with this Monte Carlo is we simulated more than almost 10,000 patients. So we have maybe 1,000 patients both sides, but we make simulation, because this is iteration process. So we have simulation of 10,000 patients. The more the simulation, the more powerful the outcome here. And that's what we did. And then we calculated the probability of achieving our target here. This is what we got. Okay, we still have time. Look at that. Now we found the following. If you give the fluoroquinolone versus Streptococcus pneumoniae that already harbor or has PAR-C gene, if you give Cepro 250 BD, it cannot achieve 30, the number, the magic number that we call here. This means what? Failure. Cepro will not ever work on Streptococcus pneumoniae with PAR-C. I tried 4 here just to make sure that this is, for the research point of view. Don't worry about that. Let's just focus on the theory, because this is what's proven here. So we found that 250, 500, 750 milligram, which is the maximum dose for ciprofloxacin twice daily, will not achieve the magic number here. In fact, it achieves 0. What's the message here? Ciprofloxacin is a poor drug, a garbage drug to treat Streptococcus pneumoniae, especially if it has some sort of resistance. So the take-home message here to any clinician, never, ever, ever use Cepro for any upper respiratory tract infection that especially includes Streptococcus pneumoniae. This is number one message. We proved it. We didn't guess. We didn't say, oh, this is from our experience. We now show them the data. It is 0. This is failure. It's not going to work. Now, let's look at let's look at galloxacin and Moxi and Gati. These are good drugs because it shows that when we give them in the therapeutic doses, and remember here that I made simulations for the doses and the concentrations in the serum and also in the epithelial lining fluid, because this is where the respiratory tract infections are there. They are mainly not inside the cells. They are in the ELF, which is the fluid around the cells. This is where the bacteria are almost there, and this is where the drug should go and act. So I simulated both of them. Let's say the infection is in the blood, or infection is in the lung, but in the ELF. I simulated both scenarios. And I found that for galinoxacin in serum or ELF, GEMI, or Moxi, or even Gati, all of these drugs are excellent drugs that can be used in these normal clinical doses, achieve 97% or more than that probability of killing this bacteria. So the take-home message here, if you want to kill this bacteria, use one of these drugs. Let's look at our friend here, levo, which is the standard therapy. When we have levo given at 500 milligram once daily, you have almost 50% probability of achieving the outcome. This is good enough? No. And even in the ELF, it's almost 60%. This is good enough? Nope. This means what? Levofloxacin, 500 milligram, is not going to work on the bacteria that's resistant, which is PAR-C resistant. Remember our patient? Our friend who died? He received levofloxacin, 500 milligram, and he had already exposure to Cepro for some reason. This means that what? This tells us what? That this patient had Streptococcus pneumoniae exposed to Cepro, developed PAR-C, and then when you give the levofloxacin in 500 milligram, this is a dire outcome. This patient was killed, not died. Because of ignorance, if somebody had looked at the ciprofloxacin exposure before, within last months, if there was a smart clinical pharmacist or clinical pharmacologist who understand all of the things that we talked about, he could have picked that and say, hey, stop here. This guy had ciprofloxacin before. Most probably has PAR-C. If we give him or give her 500 milligram, it will give me 50% probability of outcome. This is not enough because there is 50% probability of killing this patient. And that's what happened. Now, if we increase the dose to 750, you talk about 80 to 86, which is okay. But it's not good enough. I would go for 90 or above. So in this case, we say that levofloxacin, does not achieve the outcome that will target attainment that will kill these bacteria. If a patient comes with infection, Streptococcus pneumoniae, with previous exposure to a fluoroquinolone, I will not give him levo or cipro, for sure. For sure. For the levo, still wishy-wishy, but it depends who are going to treat. If you are treating your mother-in-law, that's fine. If it's your mother, no way. You got my point? So it is, my mother-in-law is just a wonderful woman, I'm just kidding, you know. I tried just the 1,000 for the fun of it, but there is no clinical relevance here for that 1,000. So the take-home message is clear. We have now numbers that tells us that this is the dose that we need to give in treatment of this type of resistance. Let's talk now about the second level of resistance. If it happened, this is what happened to our patient. Remember that he receives cipro and then he receives levo. Within the therapy, the MIC jumped from 4 to 16. What does this mean? It tells me that this patient started by PAR-C resistance. During therapy, developed the second level of resistance, PAR-C gyre, MIC 16. Let's see how can it work here. Nothing will work here. So if we give this patient whatever fluoroquinolone, he's going to die because nothing is work. Maybe MOXIE in ELF maybe has 87, but again, this is not convincing me that I give him. So in this case, the second message here, if you have a patient who has exposure to a fluoroquinolone, you should not start any fluoroquinolone. Or maybe you can start a fluoroquinolone with another drug. Now you have to use combination therapy. So the message is here, cipro, garbage, levo, normal dose is not effective. Maybe 750 is okay, but it's not good enough for me. I don't give it to my mom. PAR-C, the MOXIE, GAMI, GATI are okay, are good for me. But it depends if it's still the first level, but if it will go to the second level, PAR-C gyre, no way, no ciprofloxacin, no MOXIE, no GATI, nothing. And even to be more safe, it's advised, and now these are the guidelines built on this research. The guidelines now is that if you have to check for patient records and ask the patient, what did you receive in the last month? If someone tells you that this is a fluoroquinolone, don't start any fluoroquinolone therapy. This is going to be a real dangerous situation. So this is another message here. In this case, if you want to start fluoroquinolone, you at least have to give some sort of combination therapy. This work actually was done with a group of my real colleagues in the lab and in the simulation stuff, and these are the great guys. These are my friends and my colleagues who work with me in my lab, and I call them the winning team, and they are great guys. Thank you again.
Well, do we have any questions? Well, no questions. Now we have a lunch break.
There's one question.
Oh, one question. Well, okay.
Thank you for your brief introduction, brief lecture. Just as per our national antibiotic guidelines in Malaysia, currently, which we shifted from this, all the amoxicillin, ampicillin to community acute pneumonia for macrolides. Is it okay? We need to revise the things which fluoroquinolone's dosage increase, or we can continue with these macrolides for this community acute pneumonia?
You know, you talk about macrolide shifting because you are covering also for the atypical.
Because even I have the certain controversial with macrolides, but even this is given in national antibiotic guidelines, which we can't cross over the conditions which we practice in this situation of our government. That's only I want to ask. How we can revise this?
Simply by looking at the data and the literature. And this type of publications are very helpful. So the guidelines are built based on meeting of the big guys, the big
yes,
brains,
absolutely.
But you look at, you don't just come up with their guideline. You have to look at the literature, what happened on the real floor there. So for, let's say, for community-acquired pneumonia, not treated in the hospital, let's say in the community, simply you can have the macrolide or simply beta-lactam. If this is a severe case, PSA 3 or 4, now you have to admit this patient. In this case, you have to be more aggressive, and the guideline, the IDSC guideline, is to use fluoroquinolone, or even more aggressive therapy there based on the situation there. Or you might even use the macrolide plus a second generation cephalosporin.
Absolutely.
So it depends on setting here. So you have choices here. The message here is that, by the way, fluoroquinolones are still very effective and very good drugs, but Cipro, forget it, for sure.
Yes.
As I showed here, when you talk about the other drugs, that Gatti, Moxie, and all of these things, I showed here that they are still okay. Making sure that this patient did not receive a fluoroquinolone before. So the guidelines, I think it should be in the guidelines that simply you check with the patient. If there is previous exposure to fluoroquinolone, avoid fluoroquinolone. If there is no previous exposure, now we can test drive the Levo or the Moxie, and better Moxie or Gatti and Jemmy. You have to be careful with Levo because you have to give Levo at least high dose. There is nothing called Levo 500 milligram anymore. But based on this research, I did another research also with the acquired pneumonia, and we proved that 500 milligram levofloxacin is not going to work, and now it's not going to work. You have all the guidelines of, even for UTI, is 750, nothing 500 right now. We proved that, and by the way, this guideline based on this type of research that we did. We showed them that it does not work, it will work, and it proved clinically it's not going to work.
In your clinical, levofloxacin, how many days they treated for your patient, 25 years old patients?
You mean days? How many days?
Yeah.
Simply, the therapy for levofloxacin, if this is a successful therapy, you see in this patient, within 4 days he died. But this was a special situation. But normally, the guidelines for treatment of Levo for CAP took about 3 days. Maybe you start with IV and then send home for the rest of the week, and that's it. Some literature say that even 5 days will be enough to start and finish the therapy for that. Okay?
I doubt. Thank you very much.
You're welcome. Thank you. I thank my friend for this type of introduction. And again, my name is Ayman. iPhone, iPad, Ayman, not Ayman. Just for a reminder. Amen. So that's why you call me like your wife. Okay, that's good. It's my honor. All right, let me see what I have here. Okay. So I think I have maybe 25 minutes. And I have to say that I will have to stop after that because I'm losing my voice. So I'm not going even to, finish with Dr. Jalaf about the rest of the TDM and the best dose things. And he is the expert, so I'm not going to have anything in between. So I will just introduce the concept of the TDM for the individualized drug therapy. And I have to say that, it's my pleasure and honor to work together with Dr. Roger Jalaf. He's a great guy, an icon, TDM and pharmacometrics and pharmacokinetics, a clinician and specialist for years and years and years. And I really feel blessed and honored that I'm really attached to this big name, which is big. For me, it's a big thing. For me, I'm going just to make some sort of, laying the ground and introduce you to the concepts. And then Dr. Jalaf will begin to get you the real things about the best dose, about the metrics, and the whole heavy-duty stuff. So bear with me for the next 25 minutes, and I'll try to make it as simple as I can. Simply, as pharmacists or as clinicians, what our real goal, our goal at the end of the day is to give our patients the right drug, the right indication, for the right patient, at the right time, and the right dose. This is the ultimate goal of what we want to do day and night. Just to get these things right is our objective. And I think the right drug for the right indication, right patient, right time, this is normal practice. All of you guys, all of us are really used to have these things, and we are taking care of all of the things. This is part of our pharmaceutical care responsibilities. What I'm going to do today, which is my real interest and my passion, is to make sure that we give the patients the right dose, i.e. optimizing therapy. And when I say the right dose, I mean that the specific personalized dose for that patient, because one size does not fit all. And this is a problem for our clinical practice, that it's always one size fits all. You just give the dose and that's it. You don't know exactly what's this patient, age, physiology, what happened to the renal function, what's happening to the liver function, what's happening to the immune system, what's happening to the physiology, the blood pressure, the circulation, everything, drug interactions, other drugs there, food interaction. This is a very complicated process, and we cannot simplify it by just sampling, hey, give 500 milligram twice daily, or give 10 milligram BID, and that's it. This is a major problem in our clinical practice as clinical pharmacists. The pharmacotherapy is wishy-washy therapy. And the physicians, basically, they tend to have the overall guidelines just as they follow, and there is no real attention to the patient condition. And that's why we are trying to push the term for individualized medicine, which is very important, to make sure that we give that drug at that dose that's adequate for that patient, not for me, not for you, not for this person, because each one of us has different, reaction to the drug, different response, and at least for sure, well-documented different kinetics based on different factors. So we have to pay attention to therapy per patient, and this is very important. So the way we treat patients
is simply to identify the objective, the goal, what I want to do. I want to decrease the blood pressure to that level, or I want to treat hypercholesteremia. I want to decrease cholesterol, to that level. I want to kill this, bacteria. I want to do, this is the objective. And then I choose the drug and the dosage regimen, which is very important. This is the most important component here. But do I stop here? No, I don't stop here. I have to monitor therapy, especially for toxic drugs or drugs with narrow therapeutic window. So I monitor the response and observe for any adverse effects or toxicity, and then come back and work within the kinetics and the dynamics and the kinetics, and maybe I can change my objective, I can tailor my therapy based on what I find. So that's why continuous monitoring of therapy is very important to make it a successful process. I can give you examples of different TDM therapeutic monitoring, just simple things, just simply by clinical response. You just watch for furosemide, which is basic for, I'm going to give just an example, heart failure. So if you have increased urea and dehydration, decrease the dose. If you have edema, you just increase the dose and observe for toxic effect and toxicity or hypotension. So it's just simple monitoring for the clinical outcome, and you can change accordingly. This is still not really scientific, but this is the things that are happening all the time. This is by clinical response. The second approach is just simply to monitor through, in vitro testing. So, for example, in warfarin, you watch for the INR, and this is for thromboembolic disease. And if the INR are high, decrease the dose. If it's low, increase the dose, and try to watch for bleeding. This is very important, and so on. This is an example of in vitro testing. You just take some blood level and try to identify some markers for the therapy. And in this case, very obvious that the INR is the one here. And we go on with this thing. For the thyroxine, it's TSH. For statin, you just talk for ST. All the liver function things are important here. So this is simple, straightforward. But again, for some conditions, when you have drugs that are really toxic, and I call it, this is simply narrow therapeutic window. So you can have a window of therapy is just narrow, that below that is not going to be effective, above that is going to be toxic. So you are really working on a very risky situation here. You have to be careful. You can't be wishy-washy therapy here. You can't adjust the dose by saying, okay, increase the dose, decrease the dose, and that's it. You can't make some sort of estimate and assumption. You have to have numbers, you have to monitor, to have calculations, and you have to make sure that you are giving the drug within the therapeutic window. This is very important. Otherwise, the patient either will be subtherapeutic, it's not going to have an effect, or it's going to go to toxicity, and sometimes it's fetal toxicity. So it's very important to have close monitoring, and this is what we talked about, which is basically close monitoring of drug therapy. We just basically take drug levels quantitatively and try to correlate that with therapeutics and toxic effect. And this is very important, because if you miss something here, if you screw something here, you are going to kill the patient. This is very important, especially for drugs that are really toxic, with the therapy there, like, aminoglycosides, for example, like, phenytoin, like lithium, theophylline. If you don't adjust the dose within the therapeutic window, you are going to run into serious troubles with that patient and might be fatal at one stage. And that's why it's very important. It's very important to emphasize TDM, especially for all of these dangerous drugs. Now there is some new trends now even to extend it more beyond that to maybe chemotherapeutic agents, because they are still very toxic, very dangerous drugs. But you have to monitor that to make sure that you really give them the right dose to this patient. And again, one dose does not fit everybody. One size does not fit everybody. You have to specify the dose for that patient. That's what Dr. Jellis is going to talk about today, that the tools he is using to make sure that you give it to the right patient with this type of condition, with this type of physiology, with type of renal function, with this level of toxicity. You make sure that you tailor therapy to your patient need, patient by patient. You don't just assume that 10 milligram once daily it will be given to them, and that's it. No, you have to have close monitoring, you have to have levels, do the calculations, do the math, understand everything about the patient, understand everything about drug interaction and other medication, other therapy, and then begin to come up with the real dose for this patient. This is very important. And this is an example of some drugs that have low therapeutic index. I call it, a narrow therapeutic window, and these guys are having some sort of variable kinetics. When you have a drug that have saturable elimination, very dangerous. If there are also some sort of genetic factors like the poor metabolizers, and this is now when we have the pharmacogenomic science is starting to evolve right now, and if you have different drugs, polypharmacy, you have different drugs at the same time that are interacting that can increase or decrease the metabolism or elimination of the patient. Now we have serious condition of increased concentration to the toxic level or decrease it to less than therapeutic, the subtherapeutic. And this is when you have to dig in and have close monitoring. That's why we call it therapeutic drug monitoring. I'm not going to talk about that. This is a description. I'm going to give you just simple example here. I'm an antibiotics person. So I'm going to give you our story, the way we try to do it, in the antibiotics field. How would we develop some science to make sure that we identify different factors in antimicrobial therapy? And then how did we come up with some sort of models to make sure that we give the right dose to the right patient? Dr. Jelif will take over after that to give you the broader picture and give you more advanced and sophisticated tools, including the best dose, and hopefully can talk also about the P metrics to give you the whole picture and the tool that will help you to understand that concept. But I will focus here on the antibiotics in my business, my little shop, what we do in the lab to try to identify these issues. So again, as I talked about it before, we try to make marriage between kinetics and dynamics. And again, we have all of this stuff that I talked about it, that you make sure that you get some identified parameters to identify the outcome. So now we know that we have to give this dose to treat this type of resistance, and now we are quite sure that we are giving the right dose for that patient who is infected with that organism, with that level of resistance. This is more specific type of therapy. And now we end up with new theories. In fact, there is new classification of antimicrobials right now. And where you have some parameters that can indicate efficacy. For example, the parameter that we reach here is that the concentration, which is the peak above the MIC, this is one very important parameter. The time the drug spends above the MIC, another very important parameter. Area under the curve, above the MIC, another very important parameter. And again, as I said all the time, when you talk about AUC or concentration, you talk about the free drug. So you have to calculate for the protein binding here. Then there are some other sub-parameters like the post-antibiotic effect, persistence effect, the sub-MIC, post-exposure, all of these things, or even the immune system, post-antibiotic affected by the leukocytes. These are facts that I have to consider. But for sure, these are the PKPD principles or parameters that we have to focus to make sure that we can tailor the therapy or the dose of the antibiotic to that patient with that kinetics, with that infection, with that organism, with that resistance level. This is very specific approach to make sure that we can have good effect. So now, at the end of the day, we ended up with 3 categories of antibiotics. In fact, I'm going to divide them just to summarize to 2 only because the third in between made lots of confusion. So we just, let's say 2 now, right now. We talk about antibiotics that are time-dependent killers. There is no effect of the concentration or the amount of the drug you give. The example here are the beta-lactams, like cephalosporins, like the penicillins, meropenem, clindamycin, oxazolidines, some macrolides, but this is again now changed to the other side, flucytosine. But what's your goal here? Your goal is to increase the
or optimize the duration of exposure. You don't have to worry about the amount of the drug, i.e., let me say a patient was given amoxicillin 500 milligram Q12 hours, every 12 hours. And it was found not to be effective. The patient is not really recovering. And you want to optimize the dose. In this case, because this is a beta-lactam, what you do? You increase the dose. Let's say, let me tell them, make it 750 to 12 hours, or give the patient 1,000 milligram to 12 hours. This might be a choice. No. Based on our rule here, this is a time-dependent killer, i.e., the only factor that will really judge the outcome is the duration it extends or extends above the MIC. I.e., you need to make sure that when you give the drug Q12, you need to make sure that you have a concentration above the MIC as long as you can. How can you do that? By increasing the frequency of therapy. So you decrease the time. So other than giving the amoxicillin Q12 hours, give it Q8 hours. Or give it the same dose, or give it Q of every 6 hours. This way, you give one dose. Before it goes down below the MIC, you give the second dose. Before it goes down the MIC, you give the third dose. Then the fourth dose. So once you give it Q6 hours, you are sure that the beta-lactam is spending longer time above the MIC. You don't have to change the amount of the drug. And this is a very important factor because, hey, if you look at your practice site, you'll find that most of the clinicians and the clinical pharmacists, when you have amoxicillin every 12 hours, so it doesn't work perfect. Give them 1,000 to 12 hours. Increase the dose. Just give them more drug. And this is absolutely wrong. It is better to keep the same dose, but give it more frequent, Q6 hours or Q8 hours. This is the outcome of what we did. We proved that those drugs, like the beta-lactam, clindamycin, oxazolidones, macrolides, some of them, this is not all of them are time-dependent killers. It depends on the time they spend above the MIC. And your goal is to optimize the duration of exposure of the drug. All right. The other group, I removed the second one because it's basically between wish and gut. Now it's so proven that it's just only two. So the other group, we call them concentration dependent killers, i.e., simply their killing effect depends on the amount of the drug and the level or the Cmax or the concentration it achieves in the blood. The more you give, the better effect. The higher the dose, the better the effect. Higher the concentration that you achieve, the better the effect. Examples, aminoglycosides, like gentamicin and amikacin, all of these guys. Tobramycin, all of these guys. Fluoroquinolones, right? Levofloxacin, Cepro, and all the ones. Daptomycin, ketolides, and amphotericin. In this case, what's your goal? Is to optimize concentration and amount of the drug, i.e., you increase the amount of the drug, increase the dose as much as you can. Of course, you don't exceed to the toxic level, don't give mega dose, but you can increase the dose, double the dose, whatever. But you increase the amount of the drug. This will increase area under the curve, 1 point. When you increase the amount of the drug divided by the BD, it will be the Cmax. You increase the Cmax or the concentration of the drug that you achieve. So other than achieving 10 microgram per ml, you achieve 15 or 20 or whatever. The more you increase the concentration, the more you increase the amount, the better the efficacy and the effect and the outcomes. And this is for this type of the antibiotics. And we are always worried about the peak Cmax and also worried about the AUC compared to the MIC. This is very important. That's why, do you know about the single dose glycoside? It's based on the same theory. And it was, I think, introduced by David Beckel in Harvard Hospital,
Korea at the time, that they thought about it and said, hey, we give gentamicin 80 milligram Q8 hours, and we try to follow the kinetics. But this aminoglycoside is a concentration dependent, so it's better to increase the dose. So what if we give it a mega dose, increasing but other than 80, we just make it, let's say, 450 once daily. So you reach a peak, high peak of the aminoglycoside. Once you reach high peak, you need what? More killing, quicker killing, better killing, faster killing. At the same time, when you leave it to go down to the trough, you go to the second dose, the second day, you are still having the effect of the drug, because this is a concentration killer, and it has also what we call it post-antibiotic effect. So there is no problem if you get it to lower than the trough, and you get it to the second dose, and so on. And it was proven that at least it is less, it's equal to the conventional dose, and at least it is not more toxic. So some theories, it's not proven yet that it has less ototoxicity, and some theories that it has less theory on that. I'm not going to go about that. But at least you achieve the same effect. I'm arguing again that I know that it's more than the same effect. But at least think about this, other than giving Q8 hours. Nurses have to give 8 hours and 3 injections or 3 infusion every 8 hours. We just get 1 injection, 1 infusion every 24 hours. So at least from the economic point of view, we are achieving something. And from the clinical point of view, based on the theories here, it is more effective and less toxic. So this is why you have now this single dose aminoglycoside, once daily dose aminoglycoside. You have now extended infusion of meropenem, with even continuous infusion of meropenem. Essentially, meropenem is a it is not position dependent. It is basically, time dependent. So what had happened is that you want to keep the concentration above the MIC. So other than giving meropenem Q8 hours, give meropenem as a continuous infusion. So this way, you have higher than MIC, keep the infusion for 24 hours. You are sure that it is above the MIC. So we use all of these principles to optimize therapy for the patients. All right, so how we achieved this type of model, I'm going to go over that quickly. We did some lab works, we did some animal works, and then our clinical simulation that I showed you, the Monte Carlo, all of the things here. For example, the lab work, this is a model it's in almost most of our PKPD guys like me and my friends, George Zanel, George Rosano. This is simple, 1 compartment model, and this is the central compartment where you have like the blood, and you have the bacteria growing here, and you have the blood. It's basically a broth like a blood, and there is a pump that can pump the blood into the central compartment, and then the pump that can get it out of the central compartment. This is typically a model for antimicrobial effect. When you have the antibiotic added here, and you take samples, you can now follow the growth and kill for the bacteria in the central compartment. And this is a picture of the model here. Another model here that I built, I called it shish kebab, because I like it. It's basically the same thing, but we have more advanced model here, because we have modeling a biofilm infection, not just simple infection in the blood. It's basically the chronic infection in prosthetic valve, for example, or in chronic infections, in the wound chronic infection, maybe in the bone infection. So we model that, and we have copies here, and we grow the biofilm bacteria here, and then we run the blood here, and these are the 2 pumps here from the main compartment to the central compartment, and then to the outlet compartment. And then we take samples over time and try to calculate the outcome of therapy when we add antimicrobial to the central compartment. So this is basically a modeling that shows us what happened in vitro when we have different doses and we have different bacteria. So we model what happens in the real life there. Another sophisticated model, which is the tissue culture, the
fiber, hollow fiber tissue culture bioreactor model. And here we have nanofibers here and very small pores in between where we can grow cells. And I use this hollow fiber, by the way, for cancer therapy. So because I grow cells here, I grow cancer cells. For example, we have experiments with growing glioma cells in a 3-dimension. And then again, we have pumping of the, like the blood and then the outlet here. And here we can have the addition of the drug or the chemotherapeutic agent with a pump, so we can model the therapy of the drug, and then we can have samples and follow up the growth and kill of the cells at the same time. I use that model also to grow epithelial cells like kidney lining cells, lining epithelial cells for the kidney and even for the lung. And then I infected it with Pseudomonas aeruginosa, and then we began to give the drug and see what happens if we have chronic infection like cystic fibrosis in this model. So this is a very helpful model. All of these models help you to get data to show what happens in the clinical setting and what are the principles that are affecting the outcome of therapy. So we can know how can we tailor therapy for specific condition. And we can play with all the conditions based on what we want. We can change the flow rate here, so we can have different kinetics. We can have different factors, adding drug A, drug B, maybe macrophages, so we can try to simulate the human body in this case. And this is an example of outcome here when I use it for Streptococcus pneumoniae that have MEF and ERM genes. And I gave Clary for 500 milligram BID, and this was over 2 days. And we have, this is the FUE for 10 to the 6th and 5th or 4th and 2nd. And then for the control here, the bacteria grows non-stable and then stays plateaued. But when we give the clarithromycin, we could see what will happen if we give a dose of 500 milligram BID. Is going to decrease the bacterial load to almost 1/2 within 6 hours. So this tells me that when you give this dose, you can have a rapid killing of bacteria, and you can achieve full killing of the bacteria within 1 day. And it shows me what happens if you give this dose. If I change the drug, if I change the bug, I can play with all of these things to get the right dose for the patient under that type of condition. Some other people, I don't use that much, animal models. Simply, it will help us to have large amount of doses that can dissociate different kinetics here. And this is a model here that showed the time above the MIC for cefotaxime when you give it and to the rabbit murine pneumonia model. And these are the log CFU, or the colony-forming unit for the bacteria, which is Klebsiella pneumonia. And in this case, you notice that the time above the MIC and the bacteria killing, they are having some sort of lining here with the R square is almost 94%. So there is lots of association between those things. This shows that clearly that the time above the MIC is very important factor to help killing bacteria with cefotaxime. If it was something other than cefotaxime, it will be scattered all the place because this is not a time killer. It is concentration killer, so it will not be as organized line like this one. The last thing that I did, I showed you before, which is simply Monte Carlo simulation, where you have the PK/PD model into a Monte Carlo model. And again, this is the example that I gave it to you before for the Streptococcus pneumoniae, for fluoroquinolone, and these are the conclusions. This way, we could show that when you justify the dose, when you personalize the dose to the patient in that condition, with that kinetics, with that dose, with that infection, with that bacteria, with that resistance pattern, with that gene that's causing this resistance, we could focus and try to find the best dose. And this is what we call personalized dose in this condition. We can identify the exact dose for that patient under this condition. Thank you very much. And now I have the stage to Dr. Roger Joliffe to take over and give you the real business, the real things that he is doing right now with the best dose and the p-metric things.
May I ask a question?
Sure.
How do you measure the toxicity?
For what?
No, you see that you.
For aminoglycoside, for example.
Yeah.
Yeah, just try to identify the toxicity for aminoglycoside. Simply follow the renal function. Let's say creatinine clearance. Let's say BUN. All of these factors that are really factors that will show that there is nephrotoxicity from this drug.
Okay, so you don't use any of this NIC scale to measure a level of toxicity? Or?
Yeah, this is the scale here. This is for general toxicity for any drug.
Yeah.
But I'm talking about more specialized toxicity for those drugs that are well identified.
Okay,
for example,
so more related to laboratory measurements.
Exactly. So you know that aminoglycoside causes nephrotoxicity. So you measure renal function, renal creatinine clearance. All of these things are important there. Okay? Thank you.
Okay. Let's start by talking about a famous Spanish guitar player, flamenco guitar player, Carlos Montoya. And he's famous, and he's explosive, and he's good, and he's the master of his music. But in my opinion, he does not have the ability to follow a woman as she dances, which is the key to it all. And the woman is the patient. Think of it in another point of view. One time I was at Disney Hall, and here's this really nice Russian lady playing the piano, and here's the conductor and the orchestra. And the lady is the soloist. The conductor controls the orchestra, but he follows her and suits the whole behavior of the orchestra to her needs and her movements at the time. That's what individualized therapy is about. We will see that there's a whole bunch of mathematical techniques that are developed to track the behavior of serum drug concentrations in patients and to put them where we would like to put them, where we think a patient should have certain drug concentrations at certain times. And a lot of what we have done does not come from the biomedical community, but it comes from the aerospace industry and from the military. And it uses the same approach to track enemy fighters taking evasive action, just like you track the lady as she dances, so that you can shoot them down the best, so that you can use the dose as the gun to hit most precisely the serum level that you want the patient to have. So those are the basic techniques that we have been using. This evolved over many years. But with digoxin and lidocaine and the aminoglycosides and vancomycin and busulfan and methotrexate, there's our lab. Alan Shumitsky is professor of mathematics at USC, and David Bayard works in the guidance and control section at the Jet Propulsion Laboratories in Pasadena, where they manage all the unmanned spacecraft. And here is Michael Neely, who is our new director, and Ida Bustad makes everything happen, and Mike Van Gilder is our main scientific programmer. And so for digoxin, a number of years ago, many years ago, before serum assays were developed, we were able to do 90 evaluations in 84 patients who were on intuitively managed therapy at the time, and 36% were toxic, with 2 deaths. And this was the calculated total concentration of drug in the patient's body. The survivors on our regimens had less, and they had much less toxicity, and 1 death, but a very significant reduction. And another prospective group had only 1 episode of toxicity, no deaths, and this is more the concentration that's associated with therapeutic serum levels now. And if you look at it from the point of view of a physician in practice in a community hospital, you might say that the intuitive therapy group, who had this much, had this many manifestations, and let's round that up to 2 episodes per patient per year. And that would be a good deal. And here the survivors had a whole lot less, and the prospective group also had a whole lot less. So that if you were a community hospital physician with a standing caseload of 6 patients in the hospital at any time, you would have, let's say, 2 times 6 patients is 12 episodes a year, 1 a month, And you would say, oh yeah, I see it now and then. Not realizing that you can really make it a much rarer and infrequent thing. So that's digoxin. And for lidocaine, we were able to take the incidence of conventional therapy with this number of patients with ventricular fibrillation to only this. And these were the only two who required more lidocaine, compared with 33 in the intuitive group. And so this is a significant improvement also in therapy. And for the aminoglycosides, here we see the predicted and measured serum concentrations in patients in our respiratory ICU in County Hospital a number of years ago. Then here we see the predicted concentrations here in France in Dr. Pascal Mayer's hands. And here they are after fitting the first cluster. This is predicting the second cluster of levels based on here not fitting and here on fitting the data of the first cluster. And so this shows us then that monitoring serum levels this way really significantly improves our ability to achieve the serum levels that we would like them to have. Sander Vinks at the University of Cincinnati, when he was back in Holland in 1999, had that many patients, and this many had significant infections on admission. The peaks were higher than in the ordinary TDM. The ordinary TDM was the physician got the data and used his judgment in the therapy. Here, in the actively treated one, software was used to guide the therapy. And the troughs were lower. And the mortality was somewhat less in the overall group, but the mortality was significantly less in those with significant infections on admission. And that's one of the nicest demonstrations of the use of therapeutic drug monitoring coupled with software to individualize dosage regimens. The actively treated group had less nephrotoxicity. Hospital stay was shorter by about 6 days, with a significant saving in money. For vancomycin, here we see the usual linear regression on the logs of the serum concentrations versus a two-compartment model and Bayesian fitting. Much better ability to predict the second cluster based on fitting the first. And if you want to give it continuously IV, it's a big improvement also, because if you want to get a trough of 12, you should give 4 grams a day if you're going to give it once a day, 2 grams, 2 grams every 12 hours here. Here is progressively less and less and less, and here for continuous IV, you're down to 1 gram a day. And that's a big saving in the exposure of the patient to a potentially toxic drug. And it does as well as the intermittent therapy. For busulfan, Nathalie Blazak in Lyon in France had that many patients. Veno-occlusive disease was significantly less in the actively treated group. Graft failure was 0. Survival was significantly better. And here we see with cyclosporin in pediatric bone marrow transplantation that there was no basic dose-effect relationship, but there was a relationship between the trough levels of cyclosporin and the grades of graft versus host disease. And they found good target blood concentrations specific to each type of graft and each kid's problem. And so the empirical strategy was to change the doses if the serum levels are significantly different, and oftentimes more than a week is needed before finding the right regimen. They used their pharmacokinetic models and our older version of our software and compared their Bayesian monitoring with empirical monitoring gathered from an examination of the other activities in France. And they looked at the costs, and they found in 85 children from their place, and they compared that with 100 papers and wound up with studies of at least 50, these were the search criteria that they used. And they wound up with 9 studies to compare with theirs. And they looked at they found that the cost of one episode of severe graft versus host disease added 102,000 euros for each patient. And the cost of carrying that out were this number divided by 3.7, so about 30. So they wound up saving about 70,000 euros for each episode avoided in that situation. And here she says that in April of this past year, that the French Agency of Biomedicine released their data of pediatric bone marrow transplantation, and their people were the best center in France with the lowest transplantation-related mortality and the highest one-year survival. And obviously it was a teamwork, but she says that the contribution of the drug dosage regimen's individualization was clear for busulfan, antibiotics, voriconazole, antivirals, and immunosuppressives. And they have the best disease-free survival in the world for children transplanted for leukemia, thanks to Bayesian monitoring of cyclosporin, and they have another paper out about that. So she says, thanks to our guys for individualizing the doses like this and giving them the tools. Anders Asperg in Oslo at the Rikshospital, where they do all the kidney transplants for Norway, has shown that the use of the best dose software did better than their experienced transplant physicians in optimizing the doses of tacrolimus. And they also showed that it was useful to include the genetic information in the population model, but after you had taken 3 or 4 serum concentrations, you had learned everything that you would have known anyway. And it really pays to look at the behavior of the drug itself so that you will see the effect of all the genetic abnormalities that we have discovered and all the ones that we haven't yet discovered, and all the drug interactions that we know of and all the ones that we do not yet know about. It really is useful to look at the actual behavior of the drug itself. And Bayes' theorem describes the relationship between the prior probabilities of something, getting new information, and the way that new information revises our probabilities after we know that. And that's what the software is designed to do, and we try to do it optimally. Now, many people use parametric models and use maximum a posteriori probability Bayesian analysis. And it's good. And it fits over the entire dosage history. You don't need to wait for a steady state, which is a big plus. You can add new data as it becomes available. It's not limited to one compartment models. You can handle the correct description of the assay credibility. And the data can be at any time, not just post-distributional. And it also supplements it with what we know from past experience in the form of a population model. And it does not need one point for each parameter to be fitted. And we can fit with only one data point if needed. The results are not as good. More information does better, always. It tends to be a better predictor than non-linear least squares. Here's the Reverend Thomas Bayes, who died in 1761. And it's his work from this Presbyterian minister in Anglican England who developed this theorem that, not Pascal, the other guy whose name begins with P, I can't remember, gave him the credit for. And here we see that we are minimizing the difference between the observed concentrations and those predicted over the variance of each observation, plus the observed difference between the parameter values in the model versus the population over the variance of the population parameters. And so we start with our population model, we get our serum concentrations, we optimize our objective function to make an individual model, and then we look at the patient and we think, and we set a goal, and then we calculate the doses to achieve that goal most precisely. This is in a parametric form. We now do it in a non-parametric form, which lets you do this specifically with maximum precision. And here we see what we saw before, and again, what we saw before. Now, the limitations of that are that you assume that the probability distributions in the population have normal or log normal, for example, parameter distributions. And the action taken is only based on the central tendency and not on the full parameter distribution. And there is this thing called the separation principle. And it says that whenever you separate the process of controlling a system, any system, shooting down a plane, into first getting the best single point parameter estimates and then using those to control the system to hit the target, you do it suboptimally because there's no performance index like precision that is specifically optimized. Now let's go to non-parametric modeling approaches. And we talk about the variability now between people. And usually the parametric people describe it with a single number. But you can have specific subpopulations, fast and slow metabolizers in different proportions. And it's very difficult, it's impossible, to describe all that with a single number. And the variability within a patient over time is the assay error pattern, the error of the assay, plus the errors in recording the times that the doses are given and the samples drawn, the errors in the actual size of each dose given, the errors in recording the dosage times, the misspecification of the mathematical model that we are using, and unrecognized changes in parameter values during the data analysis. And we'll come back to that later on. Here we see the assay errors. And here we see that the assay standard deviation is the important thing, not the coefficient of variation that the clinical labs all persist in using. They close their eyes to anything in any statistics book, and they continue to do it in their really, I'm sorry to say, silly way. But here you can get the correct measure of credibility of an assay and use that so that you get the right parameter values in your model so that you can give the best dose. Here you see a population model, and the parameters are forced into this so-called normal curve. Here we are. It looks very scientific, but this is more real life. And our problem is to describe that kind of uncertainty and that kind of data. And what would be the ideal population model that you could ever make? It would be somehow to know each subject's model parameter values exactly. Pick up the red phone and call God, and he will tell you the exact parameter values for each patient. And that would be the best you could do, and you'd have one model for each subject. And the usual statistical summaries based on normal parameter distributions would lose you information from the richness of the discrete distribution that you now have achieved. And the best approach is the non-parametric approach based on these guys' theorems. And here's an example from Alain Mallet in Paris, who's really the father of non-parametric population modeling in the early 1980s. And here we see that the size of the dot, this might be the volume of the rate constant for elimination, and this is the volume of distribution, and the size of the dot reflects the probability of that point in the population. And so here we see that this is not a normal distribution at all along this axis. But this is the problem that you have in real life of finding the dosage recommendation based on what you know about your individual patients in the past to achieve a target goal for the new patient with maximum precision. And how do we go about that? So non-parametric models get the entire maximum likelihood distribution. It's a discrete, not a continuous, but a discrete joint density. And the shape of the distribution is determined only by the data itself. So you have multiple models, up to one model set per subject, and you can discover and see unsuspected subpopulations, like we saw with the two clusters in the blue slide. And the multiple models now permit multiple predictions into the future. And so you give a dose, a test dose, a candidate regimen to each model support point, and it generates a prediction into the future weighted by the probability of that support point in the population. So now you can compare the predictions of each support point with your target. It's easy to calculate the weighted squared error of the failure of that regimen to hit your target. And you can find the regimen that specifically minimizes that failure. And that is the maximally precise regimen. It's called multiple model dosage design. Multiple model adaptive controllers are in every commercial flight control system that you fly on. And it's nice to have one sample for each model parameter estimated. Steady state trough levels are not informative. Here is an early version of our non-parametric population model using all these serum samples in the data. There it is with fewer serum samples, and there it is with only troughs, so that you see how important the data is to what you get. And when you use minimally informative data like steady state trough levels, you are not learning much about your patient. Here is a simulation, and here we have a group of patients who are fast eliminators and slow ones, and here is the truth, sampling this population randomly to get 20 subjects. Here's the truth with those subjects. So the job of population modeling now is to discover this truth. If you use parametric methods, this is what you will think is occurring. If you use the non-parametric approach, this is a much better version of reality that you can perceive. And so there are no approximations. You have the guarantee that the more subjects you study, the closer your results will get to the truth. And what about efficiency and bias and convergence and fast or slow? Here we see that Bob Leary studied this question, and he set up these simulation conditions with a one-compartment model and looked at populations of 25, 50, up to 800 with two serum concentrations with a high and a low and a 10% error. Here is the truth. Here is the perception of that truth using the non-parametric model, 70 support points for 800 subjects. Here, as you increase the number of subjects, the good guys, using exact likelihoods, get closer to the truth the more subjects you study. Here, interestingly enough, the very commonly used procedure in the non-MEM software gets worse as you study more subjects. Very interesting. Here we see the same thing. The good guys get closer to the truth. The parametric approach with the approximate likelihoods at the time gets worse. Here again, the good guys and the approximate likelihood method. And so we see that, and for the correlations, again, we see that the approximate method does very, very poorly. And the efficiency, the exact likelihood methods do a good job. The approximate likelihood method does a bad job. Here we see that the two good guys converge with the theoretical maximum approach, whereas the approximate likelihood requires many, many more samples to do an equal job. And so the consequences of using approximate likelihoods are a loss of consistency, no guarantee of the more subjects, the closer to the truth. A loss of efficiency, a reduction in the convergence rate, needing 16 times rather than 4 times the number of subjects to reduce the error by half. But it has a curse of dimensionality. And if you want to estimate parameters with the number of grid points required to estimate parameters with a 1% error is actually 10 billion. So there is a real, real problem. So what Bob Leary did was to start by solving a problem on a smaller grid. When that was solved, then they threw out extra points, added extra points near the solution, and then did it over again, and did that over and over and over again until convergence. And this was about 1,000 times faster than the previous method, and much less computational effort. And here we see that the adaptive grid is very much better than our first EM method was there. And this is what we use now, and for the last 20 years, this is what we've been using. And we see that for this kind of a problem, an 8-subject, 5-parameter Michaelis-Menten model, NPEM took 2,000 hours, while NPAG took less than 2, and so on. So, and if we look at the efficiencies, direct observation has a 100% efficiency. PEM was 76%, NPAG was 61%, and that's as it should be. And the approximate likelihoods were 29 and 23, and the first order approximation was 0.9% efficient. So that is really, really sad. So the conclusion is that these guys were consistent, and this one was pretty much. Let's go here. The bottom line here is that the non-parametric approach really does the best. And so first, you start by looking at your assay and say, how does your assay do? You quantify the errors in your assay and store them as a polynomial. Then you use the non-parametric model to get your population model, and gamma and lambda are 2 ways of modeling the non-assay components of the environment in which the study was done. And the lambda is the best because it adds rather than multiplies the error. And you can predict the precision of dosage achievement, and now you can find, using multiple model dosage design, the dose that achieves your target goals with maximum precision. Multiple model dosage design. And we've been through the separation principle. You need to use the whole distribution rather than the single point summaries. The whole distribution, the entire one. And you give a candidate regimen to each model. You predict the results with each model. You calculate the weighted squared error of the failure of the regimen to hit the target at that time. And you find the regimen that has the least weighted squared error. This is multiple model dosage design. Now, when should we get our serum levels? What's the best times? We want to get them to check what we've done, but we also want to get them so that we can learn as fast as possible how the patient, how the flamenco dancer is behaving. And we need target AUCs to be precise with good sampling strategies. Methotrexate has been a problem because all that people have been interested in is what the level is when they're thinking about giving leucovorin for rescue. And because of that, they haven't started sampling methotrexate early enough to really understand the true behavior of the drug. That has been a significant problem. Now, when it comes to, again, looking at times to get levels, people have said wait half an hour after the end of the infusion to get your so-called gentamicin or aminoglycoside peaks. No, 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 no. The peak is out of the opposite arm at the end of the IV. And you don't want to waste information by getting your peak at any other time, because the peak is what tells you most about the volume of distribution of your drug. And D-optimal experimental design has been a good method. We now have better methods also, but they're too complicated for here today. But here we see that if there is a change in the volume of distribution, the time at which that makes the biggest difference in the serum concentrations is at the peak, not down here and not down here. In the same way, if the drug is in and it's going away over time, a change in the elimination rate constant makes the biggest difference when the serum concentrations are 36% of the original peak. And so those are basically the two best times, the two D-optimal times to get serum concentrations for therapeutic drug monitoring. People don't do this much, and this is a big area where we can improve our performance with TDM by using optimally designed TDM protocols. If it's given as a fixed rate infusion, the best times are when the levels are all but 36% to a steady state, and then at the end of the study period, whenever that is. For a loading and maintenance infusion, they're at the end of the loading infusion, like with the peak, and then they're at the end of the study period, whenever that is. And if you look at lidocaine, and this was done by David D'Argenio in 1983, I think, and here we see that he found these D-optimal times to examine the behavior of this system here. And so here we see that all 800 patients, two groups, one conventionally monitored and one optimally monitored. This was the time course of the serum concentrations in all 800 patients. And here we see the perceived volume with the conventional strategy, and here with the D-optimal strategy. Look how much more precise the estimates are. Here's the elimination rate constant, the conventional, and the D-optimal. So you can learn much more information with fewer samples this way. Here is the rate constant from the central to the peripheral compartment, and from the peripheral back to the central. So when you get your levels at the maximally informative times, you do learn more. And it's useful to incorporate this into our clinical monitoring protocols for patients. They help us check the achievement of target goals. They help us get good parameter estimates so that we learn about our patients most rapidly. So don't wait for a steady state ever. Start with the very first dose and use de-optimal designs and newer ones that are coming along to optimize our policies for therapeutic drug monitoring. Now, what about the diffusion of drugs into things like endocardial vegetations or into bacteria? Let's examine that and see what happens. Let's think of an endocardial vegetation or a bacterium as an onion that has different layers in it, and we can specify the number of layers, and through each layer, the substance diffuses both in and out. And here we see the time course of serum concentrations for this particular regimen of amikacin. And here we see the predicted time course of amikacin in the center of a simulated endocardial vegetation of 0.5 centimeter. And so there they are like that. And then here, for a 1 centimeter, look how much more damped the concentrations are. And for a 2 centimeter, and look how long it takes for concentrations to reach killing concentrations in those situations. But we can calculate those, and then if you believe this stuff, we can also, and here is in the center of a little microorganism, simulated microorganism, and here we have these equations that we can use now to describe the kill of organisms. Here is the rate of change of the bugs is the growth rate constant minus the killing rate constant times the number of bugs. And the killing rate constant is a Hill model, which has this shape here. And from the Hill model, we can get the EC50 if we know the MIC of the organism. So that's a handy thing there. And we can put those together then, and here we see the calculated kill of the microorganisms on that amikacin regimen when the MIC is 8 micrograms per ml. And here we see it in the center of the 0.5 centimeter, in the center of the 0.5 centimeter endocardial vegetation. And since these concentrations are all above the MIC, we get a good kill. And here the same thing for a 1 centimeter, and here for a 2 centimeter, but look how long it takes. And here now is in the center of that little hypothetical microorganism. There's the concentrations, and there's the kill, since they're all above the MIC. Now here is the concentrations from a patient. Here we see the concentrations in the theoretical little microorganism, and now the MIC is presumed to be 2. And this is for the relationship between gentamicin and Pseudomonas from a study that Pascal Mayer did on data from William Craig in a mouse thigh model. And here we see the effect of the serum concentrations and their failure to kill the microorganisms in the serum because the drug is not there. So it's not just the peak that's related to killing, but it is also the time above the MIC, even for concentration-dependent drugs. It's the time profile. And here in the center of that little vegetation, here we see again that the bugs grow out. Here we see in the beginning the serum concentrations and the concentrations in the peripheral compartment. And we see also the very, very poor fit of this equation to the measured serum concentrations, and we'll come back to that. Because this is a highly unstable patient whose pharmacokinetic parameter values are changing during the period of data analysis. And that is the problem with all conventional fitting procedures, is that they assume that the parameter values are fixed and unchanging throughout the period of data analysis, and that's a real problem. Here we see the same thing in the center of a simulated microorganism, and there they are. And here they are again in the with regard to the bacterial kill. And again from the kill in the center of the microorganism. Now, we've had a little trip through this time, and now let's begin to review this and come around again, because it very likely has been a little new to you and strange. So let's come around again and look at this, pretty much the same things, but in a little different way, perhaps. So here's our basic model of a drug, of a patient with a gut and a serum and a non-serum effect compartment, or a non-serum compartment of the drug. And again, we're going to talk about developing maximally precise dosage regimens using non-parametric models, multiple model dosage design, correct description of assay errors, estimates of creatinine clearance from not from a single serum creatinine, but from a pair of changing serum creatinines, so that you can follow the dancer better as she dances her dance in the ICU with all her unstable, acutely ill problems. We're using optimal TDM protocols, and we're using non-parametric Bayesian adaptive control tools. And so here we go again. Again, the same thing, what's the ideal model? And the non-parametric models do the best. There is Alan Mallet's original model. And again, we see the two basic clusters of more rapid and more slowly eliminated drugs. Even if we classify them, we still have the diversity in the classified populations. So our desire is to develop a dosage regimen that is maximally precise based on the total amount of information that we have, which is multiple model dosage design. We don't want to use just therapeutic ranges, because the bottom is not the same as the top of a therapeutic range. We will do better by setting, if you don't know where you want to be, select a specific target in the middle of the therapeutic range. But don't necessarily be restricted to therapeutic ranges, because those are only where most patients, and certainly not all, do well. Don't be afraid to go outside if your patient's behavior tells you that that's where you think you need to go. I've seen patients who required a serum digoxin level of 8 in order to get adequate rate control with their atrial fibrillation. Way, way outside. Social pressures are so great to stay within the so-called therapeutic range that we forget that just like we need to adjust to the MIC, or the sensitivity of the organisms we try to kill, we need to recognize the fact that patients are probably just as variable as the organisms in their response to drugs, and that the target serum level that we select for each patient should not be simply based on what the totality of society says, but you need to go where the patient's behavior takes you. The patient will tell you where they are doing well and where they are not. Follow that. Come on, Roger, get off of this. Now, if you use the mean population values to develop a dosage regimen of IV vancomycin to achieve a target goal of 15 micrograms per ml, and at each of these dots, each of these vertical columns, is where you want to achieve your target goal of 15. If you do that and use the regimen based on the means and give that regimen to each support point from which the mean values were derived, this is the great variability that you will see. This shows you the danger of developing dosage regimens based on means, when in fact, if you use multiple model dosage design, this is what you can get. Much more precise achievement of your goal. And so the dose itself now becomes a very important tool to minimize patient variability in response around a target serum level goal. Now, the assay errors, we want to look at the standard deviation and not the percent CV. And it's easy to do just like now, but use the standard deviation, square it to get the variance, put that in the denominator to get the correct weight. The reciprocal of the variance is the correct measure of credibility for a measurement having a Gaussian distribution of its errors. And the lab guys have their head in their sands for decades, not looking at this. Too bad for them. Too bad for the patients that they don't serve optimally. You can store the error as in the form of a polynomial this way, and you find that you don't need to censor low data anymore. You will find that the lower limit of quantification is a cultural illusion brought about by the desire to perceive precision as a percent error rather than the reciprocal of the variance. Funny thing. It's an illusion. Here's an assay with a constant coefficient of variation. And here it is, of course, the standard deviation is 0 when the concentration is 0. And of course, that's not true. Then why don't the laboratories give us blanks and tell us the standard deviation of the estimate of the measurement of a blank? What's the noise associated with a blank measurement? They never do that. They never do that. All you have to do is that, so that now you can track the behavior of the drug all the way down to a 0 concentration. And the whole idea of a lower limit of quantification becomes forgotten, is unnecessary. Here is our hospital's assay for gentamicin. Here is the coefficient of variation, and it becomes infinite when the level becomes 0. Here it is with the standard deviation, finite all the way down to 0. No lower limit of quantification at all. And a better, more rigorous definition of the lower limit of detection, because you can then decide how many standard deviations above your mean you will take as some legal requirement for something to be there or not, compared to what they do now. It's really sad, the behavior of the clinical labs. Really, really sad. Now, when it comes to renal function, we know that the clearance is the urinary concentration times the urinary volume divided by the plasma or serum creatinine. We can rearrange that to say this. And so we now know that the creatinine clearance times the serum creatinine is numerically equal to, let's say, the total urinary excretion over a stated time. And here's the reference for it. And what happens if a patient has a sudden change in renal function at this time? The serum creatinine will begin to rise as renal function drops, but it will be a significant time before it reaches equilibrium out here somewhere. So if you use Cockcroft-Gault or the MDRD or any other estimate of creatinine clearance that's based on a single sample, if you take it at this time, you are seeing only that change, and you have no idea that it's actually going out here. If you take a pair, maybe here and here, you will do a better job of perceiving this change. But methods that are based only on single samples always give you an underestimate of the change, and it's too late. It's too little, too late. 2 samples is much better. And so here we can say that the change in creatinine production, or the change in the total amount, of creatinine in a patient's body over a selected time period is production minus excretion. And we can say that the change is 40% of the body weight, second creatinine minus the first, and this is production, and this is excretion. Now, we can make a daily estimate of production based on Searsback-Nielsen's data in Copenhagen. Here's the serum creatinines, here's the weight, here's creatinine clearance. And so then we see that the errors in the gold standard, and as far as I know, we're the only group that has looked at the errors in the gold standard, the 24-hour urinary creatinine determination, is 90%, 95% confidence limits of 22%, and that's the gold standard. And so here you see our estimate compared with the measurement in the renal transplant books of the patients in the renal transplant unit in our hospital. And here we see that the scatter is very similar to the scatter produced by the error in the gold standard itself. And so here we see that without a urine specimen, in the presence of changing serum creatinines, we are able to estimate creatinine clearance with a precision essentially equal to that of the gold standard. That's why we like to use this in our clinical software, because we think it does by far the better job of any other method that we know to date. We want to optimize our protocols. Don't just check to see what you've done. Don't wait for a steady state. Start with the very first dose. Use D-optimal design. Get a peak and a sample at about 1/3 of the peak where you think it's going to be. There's a new optimal design, which is better than that. But do it that way, and you will do much better. This is where the TDM community can do a much better job. And again, here again is a change in the volume of distribution causes the biggest change in the serum concentrations at the peak. So that's the best time to sample. For the elimination rate constant, it's here when the levels are 1/3 of the peak. And the population model is the Bayesian prior. We get new data from the patient. That gives us the posterior, the individualized patient model. Here we see our software's non-parametric model of gentamicin in this case. And here we see that the dots are the tops of each model support point. If we wanted a dosage regimen to get a peak of 12 micrograms per ml, this would be what the most precise regimen for that model would do, and this would show us the uncertainty in our ability to get that peak because of the patient's diversity in the population model. So that's why we need to do TDM, to minimize that diversity in our knowledge of the behavior of the patient. So we will do multiple model Bayesian posteriors. Let's not talk about that. And we'll talk about the interacting sequential Bayesian posteriors in a minute. So here we see a patient now who has been on gentamicin, 5 levels. And let's see now what this 3rd level might be. There is the posterior model. We see how different it is from the population model. And here we see the prediction of that 3rd level. And we see that we are basically much more precise in our estimation of what's going on with that patient, having used this method of Bayesian analysis. If you use map Bayesian analysis based on single parametric models, you will not usually see this estimate of the precision with which you know your patient. Now, the interacting multiple model comes about because we assume that the parameter values can change with each new data point sequentially. And it has been shown to track gentamicin and vancomycin behavior best in ICU post-cardiac surgical patients. That's the second reference here. And here we see a patient who was acutely unstable. And here we see the fit to the data. And here we see the really very poor quality of the fit to that data. When we use the interacting multiple model, well, here then is the origin of the IMM method. And the main purpose of a tracking system is the estimation of target trajectories in the controlled area and their prediction into the near future to help you aim the guns. And here's the improved fit using that approach. Now what we're going to see is that the parameter values are going to change over time as each new data point becomes available. And here's the volume of distribution changing over that time. And we're now able to track it so that we can get the Bayesian posterior at this time the best, so that we can calculate the best regimen for the near future for this acutely ill, unstable patient. And so unstable patients have changing renal function. They have a changing volume of distribution because they also have changing fluid balance. And here we come to the issue of clearance. And many people will tell you that clearance is the most physiologic pharmacokinetic parameter value that exists. And that is simply not so. Clearance commingles the volume and the rate constant because it's their product. And you lose the actual information about the contribution of each. Clearance is an unnecessary pharmacokinetic parameter. You never need to use it. You can describe everything, regardless of mechanism, without using clearance. And you will be able to keep separate the knowledge of the volume with its implications for fluid balance and the rate constant for excretion of the drug with regard to your individualization of drug therapy. And for dialysis and renal replacement, it's good to get a drug concentration and a creatinine at the beginning and end of dialysis or renal replacement or extracorporeal membrane oxygenation. And because you're getting creatinine there, you can also estimate creatinine clearance with a pair of serum creatinines so that you can then link the behavior of the apparatus to a value of apparent creatinine clearance that it gives you. So that then if you don't have the data, you can use that information to help you understand what's going on with the patient during each of those procedures, so that we can now include all of those procedures into the history of the patient's pharmacokinetic data, so that we can take better care of them that way. Now, just like bacteria with their different MICs, patients also vary in their sensitivity. And digoxin clinical effect does not correlate with the serum concentrations, but with the concentrations in this peripheral non-serum compartment. This is data from Jim Doherty a number of years ago, and here are serum concentrations in patients who are toxic and patients who are not toxic. Look at that, 6.5. Not toxic. People vary that widely in their sensitivity to drugs, just like the bacteria do. Every person has their own MIC, so to speak, their own index of sensitivity to the drug. And these patients have to have very different binding constants of the drug to their sodium potassium ATPase in order to be able to tolerate those concentrations and to derive benefit from them. Here we see the work of Dick Rooning and his colleagues at Ohio State in 1973, from which we take the data for our population model of digoxin. It's right from their data. And here we see the plasma levels decreasing from the IV administration, and we see the concentrations in the peripheral compartment increasing and going down. And here we see the measured QT interval in the EKG, which is a reflection of the inotropic effect of the drug. And we see it following the peripheral concentrations rather than the central. And so we like this two-compartment, non-parametric model with unconstrained parameter distributions. And we plan regimens either for a trough serum concentration of 1 or maybe 0.7. But patients with atrial flutter, instead of needing a peak of 7 in the peripheral compartment, they may need 9 to 18. You have to look at each patient and see each patient's response and sensitivity to the drug. And here we see data from the literature. And the one that's most often quoted is from Falk, and it says that digoxin is no better than placebo for converting patients with atrial fib to sinus rhythm. And here's another study that says the same, and here's another study that says the same. But here's another study that shows 71% conversion with more out there. And here's another study that shows 85% with more out there. And this is significantly different from here. So follow what happens in the peripheral non-serum compartment for digoxin and compare that with the patient's clinical behavior. And then decide what you want to select for a target goal in that peripheral non-serum compartment. Here's a patient with chronic stable atrial flutter for at least 3 years. And every cardiologist knows in his soul that DIG will never do anything for this kind of a patient. But this was the first patient that happened to convert, and I wasn't expecting it at all. I was simply trying to control his rate. And here he was with atrial flutter and an apical rate of 140. And here we are giving him digitoxin, and the little crosses here are the serum levels, which you don't see well, and the top line is the peripheral compartment concentrations, and the bottom here is the serum concentrations, and this is the first month. And here we were titrating him up, and here all of a sudden, sinus rhythm, right out of the blue, at 110. Here he had a little first degree, a little 3-to-1, and here a little 4-to-1 AV block, but here now, sinus rhythm. Now at this point, he aspirated and developed an aspiration pneumonia, and everything went south. Everything went down the tubes, back to atrial flutter with an apical rate of 140. We increased his DIG a little bit, but the real thing was that his aspiration pneumonia got treated. His blood gases, which had been bad, got better, and he now came back to his sinus rhythm. And he did that 3 times during this 6-week course. And 3 times when his blood gases get better, even though we didn't do anything with his DIG after that, he came back to sinus rhythm, showing the combined effects of the DIG and his blood gases on his behavior. But we were able to be useful to him this way. Here's another patient. This is a lady, a 94-year-old lady with a transitional cell carcinoma of the bladder. And here she was one Saturday afternoon when I was covering the geriatric service with new onset fast atrial fibrillation at about 180. And here her creatinine clearance was 3.7. And she had given up. She wasn't taking food anymore. She was getting ready to die. But I gave her 0.5 and then 0.25 and then 0.25 again here. And at that point, she converted. And now the task was to give a maintenance dose that was correctly related to what I had done in order to keep her peripheral compartment concentration at the level at which she had converted. So when she converted, I came back and put the data into the software, and here we see that her peripheral concentration was about 9 when she converted, and that's what we developed a regimen for after that. And it turned out to be 0.125 8 times a week. So 1 day a week she got a double dose. That's here and here. And in that way, we were able to keep her peripheral compartment concentrations in the region where she had had them when she converted, and she stayed converted for the remaining 2 weeks of her life until she died. But we were able to do that for her. Here's another patient with new onset atrial fibrillation. He was on 0.25 once a day at this point. He missed a dose here and went into fast atrial fibrillation. Here are his serum concentrations. They gave him 4 doses of 0.25 here. This was up at Stanford, and this was all by phone. And he converted to sinus rhythm, and they put him back on his old maintenance dose, and so he excreted the remaining dig and went back into atrial fibrillation here. They converted him again with only 2 doses, and again put him on his maintenance dose, and he went back into atrial fibrillation here. They converted him again here, and now the job was to come up with a dosage regimen that maintained his peripheral compartment concentrations at that level afterwards. And the dose was a big one. It was 0.5 and 0.625 milligrams a day. And very few cardiologists would be willing to do that because it's not in their experience. But Bill Nicholson, who had this patient up there, had been in our lab, and so we did that. But you don't see the thing clearly here. Look at it with his peripheral concentration there. And here we see when he went into atrial fibrillation, when he converted the first time, back to atrial fibrillation, converted the second time, back to atrial fibrillation, converted the third time, and we said, well, he's telling us that he likes it when he's got between 11 and 13 micrograms per ml in his peripheral compartment peak. So we came up with that regimen to put him there, and it did the job, and he stayed in sinus rhythm for the next 2 weeks in the clinic. Here's another patient who did not convert because they wouldn't give him what he needed. And his serum concentrations got pretty high, but his peripheral concentrations were much lower, only about 6 rather than 9 or 11 or 18. And that's why he didn't convert. And each model seems to have their own, each patient seems to have their own relationship between serum and peripheral compartment concentrations. The population model has a ratio of 7. The lady with the end-stage renal disease had that basic relationship. The patient who converted, who needed conversion 3 times, the patient at Stanford, had that ratio. The patient who did not convert had only that ratio. So it's important to look at the patient and to follow the concentration in the peripheral non-serum compartment for digoxin and to develop your therapy to hit a target in that peripheral non-serum compartment. Here's another nice article by this guy Bauman, and here is the overall mortality in the placebo group, and here in patients on digoxin. And here we see that there's a significant drop in the mortality with a serum concentration of 0.7. So that's probably a good target goal for a serum concentration. That's where I try and keep my wife. And she takes 0.125 5 days a week and does it that way. Now, the drug companies spend a lot of money developing drugs. They miss a lot of them. A lot of them don't make it. But they do prey unethically and immorally on a pharmacokinetically untrained and uneducated medical community. Look at the TV ads for the oral anticoagulants and the antiplatelet drugs. Look at them and cringe. Here's a nice article, and it says that prasugrel is better than clopidogrel. And they say because it has better outcome and more antiplatelet effect. They say nothing about the dose. Here is their publication of the antiplatelet effect. Here's the prasugrel, here's the clopidogrel. Notice that both of them cover the full spectrum from practically no effect to practically total antiplatelet effect. Do they think to ask, what is it in those who bleed? Do they think to ask, what is it in those who are poor responders? Do they think to ask, what target value could we find where patients do best with respect to outcome? No. No. And this is a well-recognized group in Italy, Dr. Antonucci. And their response to the letter to the editor that I wrote really shows the depth of their ignorance, because they say the variability is far too great, so the individualized therapy is unrealistic. Sorry, fellas. But it's the drug companies that are being unrealistic and bad guys to foist this upon us. They suppress their own data, which you see is there. And the data is there to relate each of those to outcome, and they simply did not look. And Dr. Antonucci didn't even think to look. I also asked not who sponsored the study, but who designed the study. They didn't answer the question. They only said who sponsored it. But they could have done anything, all of that. And for them to advertise that this is not needed is really, in my view, unethical behavior. And Big Pharma is like that all the way through. Big, bad pharma. You have to watch them like a hawk and try and catch them. Why do we accept that? Why do medical schools continue to teach medical students to give doses out of the package insert rather than any pharmacokinetics? There isn't a medical school that I know of that teaches pharmacokinetics in any clinically meaningful way anywhere in the world that I have seen. They suck it up from the drug companies, and they think their stuff, whatever that is gold. And again, here's our lab. There's Alan Chemetsky and Dave Bayard, our key mathematicians. Mike Neely, I'm so glad, my God, has he just taken this stuff and run with it. And his new version of Bestos that's going to come out maybe in a year or so, has the ability to model the behavior of combined multiple interacting drug systems so that you can model drug interactions in a quantitative way and then develop combination coordinated chemotherapy that manages rather than avoids those interactions. That's what's happening with him. And here we are again. So thank you very much. Any questions? Like Mr. Nixon used to say, I hope it's all crystal clear.
Thank you, Roger, for this very enchanting presentation. And I have a question because I always feel that all the time. You talked about optimal sampling time. I always feel that when we have patients who are very fragile patients, let's say children with malignancy, and you want to do some...
I'm going to say it again, I can't hear.
Yeah, if you have patients who are very fragile, like children with malignancy, who are receiving multiple doses of different drugs, and you want to do a kinetic study for one of the drugs, for example, you have hard time to schedule a dosing or sampling times and say every 6 hours or the second day, third day, fourth day. It's unethical and unpractical. So can you just please elaborate more on how can we go over that by just choosing maybe one sample, every...
That's been the attitude of a lot of people who have to take care of kids.
Yeah.
Because they don't want to draw any more blood than they have to. But you also should think about drawing as much blood as you need to for their benefit. And acutely ill people, grown up or kids, are going to need more frequent serum sampling. They just will. When they're unstable, you need more data to track the behavior and control the behavior.
But then I can use some way of...
And that's where these optimal policies for getting TDM samples are very useful.
Yeah. But then maybe I can use your technique just to optimize the optimal sampling time so I can identify when exactly I should draw the samples, and I know that this is the right target, and I can build a plan for that.
And another thing, if the doctor orders a dose to be given at 8 o'clock, That's a physical impossibility. No patient ever gets a dose at 8 o'clock because the nurses go around, and they have to go around and go from patient to patient to patient, giving them their drugs. All they need to do is to write down military time to the minute when they did these things, and then nobody cares when the drug was given, just so that we know when it was given. And that's the important thing.
Okay, thank you.
Thank you very much. I think your speech was very nice, and I really appreciate a lot of your things. Even I'm not Bayesian, I'm a Neumann-Pearson statistician, so I'm not so fond.
You are a frequentist?
No, I'm a Neumann-Pearson statistician, but I know that somebody's used these Bayesian models, but I don't think we shall discuss that here. No, but there was one thing I'm wondering, because you stated that the standard deviation was nearly linear. It was nearly that the standard deviation was was nearly linear with regards to the dosage.
Well,
but from all my
you should react to a measurement according to a correct measure of the precision of that measurement.
Yeah.
The reciprocal of the variance of a measurement is the correct measure of a measurement that has Gaussian error distribution to it. And that's why reciprocal of the variance is much more useful than CV%.
Yeah, I know that. It wasn't my question or my comments. It was that the standard deviation, if you see that standard deviation as you see that the standard deviation is increasing with the dosage. And that's correct, but I don't think it's linearly. It's following more log normal, log distribution. So it's actually flattering out. It's increasing in the beginning when you increase the dosage in the beginning. But if you increase it more, it will flatter out, and you haven't.
You're absolutely right. You're absolutely right. It's not linear, and we had that slide that shows that in that particular assay, it was a little more at the blank than it was down in here, and then it came up here like this. So you can store the information of that nonlinearity as a polynomial, so that then for any single determination, you can have a pretty good estimate of the standard deviation of the assay.
Yeah, because what I had done is that not in pharmacokinetics, but in clinical trials where we are looking into centers, and the more centers you are including, the standard deviation in the total material will increase.
I'm sorry,
but it's
more what?
The more center which you include in a clinical trial. If you,
greater than places taken,
then the standard deviation will increase, but
sure
it's not linearly. It comes to a certain point. It's following a logarithmic deviation, and then it flatter out.
Whatever it is, you can catch it with the polynomial.
I agree. But thank you anyway. I really appreciate your talk.
Thank you very much. Thank you all.